to genetically characterize it and prove it even is a virus particle, and not just the usual, normal, regular dead cell tissue debris breakdown particles from dying, rotting cells in their petri dishes. You must isolate that particle alone and apart from all of that other stuff to prove it is a virus and not just cellular debris garbage.
What do we mean by virus isolation or virus purification? These are jargon words in virology, and they're not very precise. They mean different things to different people.
No, it specifically means to remove that particle alone and apart from all of the other identical cellular garbage decay and genetically and chemically characterize it to prove it is a unique thing called a virus and not just more cellular garbage.
Isolation, what was it?
Isolation and purification.
Of the virus?
Yes.
Well, you isolate a virus by finding the virus which causes a disease. And you purify a virus by making just by purifying it so you get a pure virus.
Okay.
I don't understand what the issue.
They interchanged the two, and I wasn't sure
I see.
If it was the same thing or if it was two totally different.
No, it depends on how they used it.
Okay. Can you explain the process of HIV isolation?
Well, didn't Dr. Gallo do that? I mean, he actually isolated it. So,
Yeah.
I mean, why should I do all of this? This is all textbook stuff you're asking me.
I'm not quite sure what's behind your question about isolation.
I don't want to be your textbook. You know?
Okay.
I got other things to do.
A viral particle must fulfill defined physical and biological properties, including being a replication-competent intracellular parasite, meaning it results in identical copies of itself inside a host organism, and also capable of causing disease in a host, such as a human. However, viruses such as SARS-CoV-2 are nothing more than phantom constructs, existing only in imaginations and computer simulations. In this paradigm, cases of invented diseases like COVID-19 are nothing more than the detection of selected, tiny common genetic sequences and proteins purported, or rather pretended, to be viral. The existence of an actual virus is not required in this fallacy loop of circular reasoning, and thus, entire fictional pandemics can be built upon fictional digital creations and then falsely sustained through meaningless in vitro test tube molecular reactions. In Part 1, we'll outline some of the history of virology and its blatant pseudoscience-like failure to follow the scientific method. In Part 2, we'll examine the fraud and deception used to propagate the COVID-19 plandemic. Finally, in Part 3, we'll analyze how health institutions, mainstream media, and governments maintain the virus illusion in tandem through information control and carefully crafted narratives that parrot virology's baseless claims.
Here's the fact which every virologist agrees to, and I can even explain that a little bit. There is not one published scientific paper where any particle that you could call a virus, and remember, a virus is a thing, it's a particle, we can sort of see it under an electron microscope, even though that's problematic. But so this thing, they tell us, there's not one published paper in the scientific literature finding this thing in any biological fluid of any sick human or animal. Anywhere. And everybody agrees. All the virologists agree. They say that's not how you find a virus.
As Dr. Tom Cowan also points out in Settling the Virus Debate, identical particles that have been successfully isolated through purification, like exosomes, extracellular vesicles, apoptotic bodies, and microvesicles, have not been shown to be replication competent, infectious, and disease-causing. Hence, they cannot be said to be viruses. Additionally, the proffered evidence of viruses, through constructing and assembling alleged virus genomes like puzzle pieces and animal experiments, derives from questionable to flawed methodologies with insufficient experimental controls. As of January 2023, following extensive inquiries through Freedom of Information requests coordinated by Christine Massey, not even a single one out of 211 government health or science institutions in over 35 countries have been able to provide direct evidence of the alleged SARS-CoV-2 virus. The institutions were asked to produce any documents demonstrating the purification of SARS-CoV-2 said to have caused disease in humans via maceration of the fluid from a human said to be sick with a virus, the filtration of that fluid, and finally the use of an ultracentrifuge to find that virus, also referred to at times by some people as isolation. On many occasions, following an admission that no such evidence is held, institutions such as the New Zealand Ministry of Health then suggest that despite having no record of them, there are several examples of the virus being isolated and cultured in a laboratory setting, and then direct you to studies that have done no such thing in their methodology sections. They are always, without fail, tissue culture proxy experiments, in which the word isolation has become detached from its understood meaning, and it has never been demonstrated that any particle of the millions found in these smorgasbord tissue cultures, imaged or imagined, has the properties of a disease-causing virus. In any case, it is a distraction from the wider issue exposed by Christine's Freedom of Information requests, which is that particles claimed to be viruses can never be found in human subjects. As Dr. Stefan Lanka points out, density gradient centrifugation is the scientifically required standard technique for the demonstration of the existence of a virus. Despite the fact that this method is described in all microbiology manuals as the virus isolation technique, it is actually never applied in experiments meant to demonstrate the existence of any pathogenic viruses. The defense of virology's methodologies is obviously attempted by its own promoters, including New Zealand government and state-funded media's favored microbiologist, Susie Wiles. Her employer, the University of Auckland, is among those institutions who have now confirmed that it has not done any work relating to the purification of any COVID-19 virus, and therefore has neither found in nor isolated from any human subject the so-called virus named SARS-CoV-2. Yet this associate professor, above an assistant and below a professor, told the whole country that
the world is on fire, and I don't think people realize that. So we just need to, what our government is doing is it's listening to the experts and it's trying to keep us safe. And so we might actually get off this relatively unscathed if we all behave as they're asking us to behave. Yes. And we might end up having to put some more strict measures in the months or so to come.
She was then ordained New Zealander of the Year in 2021 for helping millions globally see past the fear and complexities of the pandemic.
The world is on fire, and I don't think people realize that.
And helping to keep us safe. In her November 2020 article, Koch's Postulates, COVID, and Misinformation Rabbit Holes, Wiles alleged that the people like Christine Massey asking for evidence of the existence of SARS-CoV-2 virus responsible for COVID-19 are specifically wording their request to rule out obtaining any evidence that the virus exists. Her article quickly went off on a tangent about Koch's postulates being unsuitable for viruses, and she thus declared them as invalid in that context. Robert Koch actually stole his postulates from Friedrich Gustav Henle, and ironically, Koch never fulfilled them himself in his own experiments. But Henle's postulates were a simple four-step way to prove a microbe caused a disease or not. You simply take a sick animal or person suspected of having a new disease like COVID-19, draw their blood, sputum, or fluids suspected of having the causative agent, and you must isolate that agent only, using first maceration in fluid, then filtration through a nanomesh screen like a strainer, to get out all other material in the fluid larger than the suspected causative agent. Then spin that in a density gradient centrifuge so all particles are in their own density band layers. Then extract only the layer with the suspected causative agent with a nanopipette, which virologists don't do. Then, if required, or should we say if even possible, create more alleged viral particles in a test tube and repeat the purification process with density gradient centrifugation, which virologists don't do. Then take that pure culture and inject, spray up the nose with an atomizer, or drop into the eyes of a healthy animal or person, and it must cause the exact same illness and symptoms as the animal or person you took the fluids from, which virologists don't do. Then you have to draw fluids from that animal and repeat the exact same procedures from the first two steps, to see if the suspected causative agent is now in that person's fluid as well, proving transmission, which virologists can't do without cheating steps 1 through 3. But then you have to do a control experiment, taking the same fluids from a healthy animal or person and repeating the exact same steps and procedures, to both check yourself and prove that you don't find the exact same results in your culture because you either cheated and didn't isolate the suspected causative agent from the thousands of other things in a fluid sample, or you cheated and didn't use a pure culture, and the results of the culture are really from the chemical toxins and foreign tissues you used in the culture itself, which virologists don't and won't do, because it will prove they are cheating. That's why they are so hell-bent on getting rid of Koch's postulates. It is strange that nowhere in her article did our associate professor Susie Wiles ever mention Rivers' postulates, which were purposely modified from Koch's postulates to include theoretical viruses, since Koch's were created for bacteria. It was even made much easier, because Rivers allowed for cultivation in unpure cultures in host cells, like monkey kidney tissue, dog epithelial cells, and even human lung cancer cells, plus toxic chemicals and antibiotics. Plus, injecting that rotting cell culture into an animal only had to produce a comparable disease and not the exact same symptoms or disease, but it is not clear on the meaning of comparable.
And I want to poll the audience. If you could just shout out, what are the symptoms of COVID-19? What? Fever. Cough. Shortness of breath. Good. Fever, cough, and difficulty breathing, right? So what symptoms did they actually have? Weight loss and slightly bristled fur. Anyone hear news reports of bristled fur in COVID patients? Weight loss also not a symptom of COVID-19 that's reported. It's a very non-specific symptom. So once again, the experiment fails on this count, that this is not the same symptoms as the disease in question.
Perhaps Wiles didn't mention Rivers' postulates because she would have to admit that these postulates have never been fulfilled either, and that virologists have spent much of the 20th century trying to identify viruses directly from sick humans without any success. Awkwardly for Wiles, the World Health Organization stated back in 2003 that with regard to SARS-CoV-1, conclusive identification of a causative agent must meet all criteria in the so-called Koch's postulate. The additional experiments needed to fulfill these criteria for SARS-CoV-1 are currently underway at a laboratory in the Netherlands. The World Health Organization removed that article from their website in 2021, obviously because those experiments failed and people were asking questions about SARS-CoV-2. Luckily, we have the Internet Archive. Here's why they failed the WHO's requirements.
Some seem to claim that this paper that appeared in The Lancet in July 2003 established that SARS-CoV-1 could indeed be implicated as the monocausal agent. But wait a minute, what actually went on in the study involving 4 poor little monkeys? Firstly, they took a sample from a single human SARS victim and produced a cellular culture in the lab through several steps. The monkeys were then infected by squirting this mixture into their tracheas, noses and eyes. On the 2nd, 4th and 6th days, the monkeys were anaesthetized with ketamine, an anesthetic known for its hallucinogenic properties. Blood samples were taken from veins in the groin and smears were obtained from the nose, mouth, throat and anus. The monkeys were all killed for autopsies after just 6 days. 3 of the monkeys became lethargic after 2 or 3 days. On the 4th day, 2 developed temporary rashes. Only 1 monkey had any breathing difficulties, while 3 showed non-advancing tissue damage to the lungs. The lymph nodes near the trachea and the spleen were larger than normal in these 3 monkeys. The other organs all appeared normal under microscopic examination. Now, there are some glaring weaknesses with this study. Firstly, the monkeys' illnesses were not typical of SARS. Only 1 monkey had breathing issues and there was no mention of any of them having a cough or fever, which are some of the key features of SARS. And none of the monkeys became critically unwell or actually died from SARS. They were all euthanized by the researchers. Secondly, the paper claimed to identify coronavirus particles in monkey tissue on electron microscopy, but many different virus-sized particles are part of the cell culture. So without particle purification, it's impossible to know exactly what they were. Now, there seems to have been a lot of confusion about isolation and purification. Purification means that we've got rid of the host cell material and culture medium and just have the alleged virions by themselves. Virions are the complete viral particle, meaning it includes its shell and everything inside. So if it's not purified, then there will be other viral-sized particles in the material, which may appear in the electron micrograph images and may also contribute other genetic fragments. Thirdly, injecting 4 mL of culture solution directly into a small animal's trachea is a way to cause a lung reaction. Given that these little primates weigh around 6 or 7 kgs, it would be the equivalent of injecting, say, 50 mL or almost 2 fluid ounces of culture solution into a human subject. Introducing that amount of material directly into the trachea has the potential to flow into the lungs and cause reactive changes and even potentially a pneumonitis. On this note, it is also unclear what conclusions you can draw from injecting viral cultures directly into an animal's upper respiratory system and then a few days later finding it in the lower respiratory system. Forgive me for being a stickler for the rules, but overall this monkey business has not helped confirm Koch's postulates as they claimed.
Wiles also appeared to be at odds with Na Zhu et al., one of the teams that first claimed to have discovered SARS-CoV-2, because they conceded that, although our study does not fulfill Koch's postulates, our analyses provide evidence implicating but not proving 2019 nCoV, later SARS-CoV-2, in the Wuhan outbreak. Additional evidence to confirm the etiologic significance of 2019 nCoV in the Wuhan outbreak include animal, monkey experiments to provide evidence of pathogenicity. Whether different virologists want to entertain the validity of Koch's postulates or not, it is simply another distraction, as the postulates require the physical isolation of a microbe, rather than assertions that one exists, through means such as computer in silico simulations, or imaging vesicles of unknown biological function, like these identical alleged SARS-CoV-2 particles found in the lungs of both COVID PCR positive and negative patients, or these alleged SARS-CoV-2 particles found in the kidneys of a COVID-negative patient, or claiming that unpurified biological toxic soups given to or injected into animals contain viruses. Wiles admits that when virologists want to isolate a virus from a sample, meaning take that virus alone and apart from the sample itself, they instead take the whole sample, or some part of it, and add it to some cells, usually ones that are relatively easy to grow in the lab. That is not isolation. Sputum, fluid, and lung tissue samples have all sorts of particles in them, including lung tissue, nasal tissue, or swab cheek or throat tissue itself, which all have their own RNA and DNA. Bacteria and microbes in the sample, which all have their own RNA and DNA. Inhaled plant pollen, which has its own RNA and DNA. Inhaled bug and food debris in the nose, mouth, and lungs, which all have their own RNA and DNA. Particulate matter from air pollution like dust, dirt, soot, smoke, lead, etc. And then they add that to some cells like monkey kidney tissue, which has its own RNA and DNA, and then add the blood from an aborted cow fetus for nutrients, which has its own RNA and DNA, and profess they can grow, find, and isolate a virus out of all of that, but can't find it in the original sample. Wiles says, and then we look to see if the cells die and or if there are any virus particles released into the liquid nutrient bath the cells are growing in. In other words, viruses can't be grown in a pure culture as described by Koch's postulates because they need a cell to grow in. Yet the virus particle would need to get into that cell in the first place somehow, grow, multiply, and shoot out of the cell by themselves, without the cell, to fly around the atmosphere and get into other people's cells to cause infection. Isn't that why we're told to wear N95 masks? To stop the actual tiny flying virus particles themselves from getting in, and not flying host cells full of virus particles?
What's more, an N95 mask is actually really good at filtering both the largest and smallest small particles. It's medium-sized small particles that are hardest for it to block.
Isn't that the point of taking a sputum, fluid, or tissue sample in the first place? Because they should be teeming with infectious replicated virus particles. If coughing or spitting that same tiny amount of fluid can allegedly infect someone else? Despite never isolating one to check, virologists postulate that virus particles are protein and lipid shells around a capsid containing DNA or RNA, and anywhere from 50 to 140 nanometers in diameter. What biologists call exosomes, or extracellular vesicles, for example, are also claimed to be a protein shell around messenger RNA and are also 30 to 150 nanometers in diameter, exactly the same as alleged viruses. And Wiles admits, yes, exosomes, more commonly referred to as vesicles, are small particles that can be separated from samples by centrifugation. But the important distinction here is that they are bits of our cells that bud off and generally require huge volumes of material to isolate them from. The same process just can't be applied to a small sample of cells from someone's nasal swab. Yet, molecular chemists find and isolate molecules smaller than just 1 nanometer, much smaller than 150 nanometer exosome vesicles or alleged virus particles every single day without problem, and do all sorts of things with them. And IBM unveiled in 2021 the world's first 2 nanometer chip technology with 2 nanometer nanosheets, which they are easily able to manufacture, find, isolate, and even attach to other nanostrips that are only 12 to 44 nanometers wide and only 75 nanometers long, which they can clearly manufacture, find, and isolate in order to attach the smaller 2 nanometer nanosheets onto them. Yet, virologists can't find this in a large sputum sample? Wiles admitted it can be done for same-sized exosomes and vesicles with a large enough sample, but then a virologist 20 times her senior said this.
We asked a guy who was 20 years, senior virologist at Wuhan Institute of Virology, 20 years head of Yale Virology Laboratory. Can you find SARS-CoV-2 in any fluid of any person you say has COVID? He said no. Why not? Because there's not enough virus to find. Okay, we asked him, what about if you put 10 people with supposed COVID and you mix their bronchial fluid together? Is there enough? No. 100 people? No. 1,000 people? No, not enough to see. 10,000 people, you mix their fluid together. Would you then have enough virus to see? And he said no, and then he wouldn't answer anymore.
Yet this minuscule amount of fluid is supposed to contain enough virus particles to make a person dangerously sick from up to 6 feet away? As Dr. Bailey points out, it is unclear if Susie Wiles is implying that isolating a virus is established by taking the entire cocktail sputum soup of various materials from a sick person, or just some part of that sputum soup, or adding either of those to foreign tissues and fluids in a test tube that have nothing to do with that sick person, or is it just seeing some foreign tissue die in a nutrient-starved and poisoned test tube? Or is it the release of the dead tissue debris claimed to be virus particles in that test tube or tissue culture, or all or some combination of these elements? In fact, nothing Susie Wiles describes even requires the existence of viruses, as Dr. Stefan Lanka's control experiments finally proved, repeating their same foreign tissue culture procedures and getting the exact same cytopathic results with no added sputum sample at all. It is all a game of deception, whether realized or not, and it is time we all see through how it is being played against us. The claims that SARS-CoV-2 has been shown to exist through this methodology are all scientifically invalid, as none of the experiments were performed with valid scientific controls. Virologists are supposed to be studying alleged viruses that spread between, attack, affect, and harm human tissues, not monkeys. So why are they using green monkey kidney tissue in these cell cultures? In fact, the issue extends beyond just SARS-CoV-2. With the exception of bacteriophages, which are very real and not viruses, every virus asserted to exist relies on similar pseudoscience. The history of virology reveals that the types of cells eventually selected for these experiments have been those that have a propensity to break down with the claim of virus-induced cytopathic effects or cell destruction. For example, Vero E6 monkey cells from the kidneys of African green monkeys have long been favored by virologists, supposedly due to their suitability to host many viruses, but suspiciously also because the aneuploid kidney line with an irregular number of chromosomes is more susceptible to toxic insults from the additional ingredients they add, such as the kidney nephrotoxic antibiotics and antifungals added to their culture mix.
So this is how they do the procedure, very different from what I described. They call this virus isolation. It's really a tissue culture experiment. And they take the fluid from the sick person that they suspect has a viral disease. They filter it very coarsely. Sometimes they do a basic centrifugation. This is not purification. It's just to remove debris that forms a pellet, but it doesn't even remove all of the cells. And And then they do a tissue culture, but they vary the conditions of the cells. They usually use foreign cells, like monkey kidney cells. They may use other foreign cells. Occasionally, they use human cells. They also use fetal cells on occasion. But they don't continue the normal culture conditions. Instead, they reduce the nutrition to 10%, generally speaking, sometimes lower of the normal amount. Then they use a modified nutrition, which has is less nutrient dense. And then what they do is they add fetal calf serum, and that, because it's from a fetal animal, it has some factors in there that improve the survival of the cells in the culture and also is a source of protein nutrition. But of course, it's a foreign material, blood product. So if there are such a thing as viral particles, it would certainly be a source of contamination. It's also been shown in the literature to be a source of contamination for any downstream genetic testing. So if they were to do a PCR test, for example, on the culture fluid, the results would be contaminated by the fetal bovine or fetal calf serum. There are papers that looked at that that basically say that the genetic testing would be worthless if you add this constituent. Then they add the antibiotics, and they do these at higher than normal concentrations, usually 2 or 3 times the normal concentration. They often pick antibiotics with nephrotoxicity like gentamicin, aminoglycosides, amphotericin, and streptomycin when they're using kidney cells. And under these conditions, they observe the culture, and they find what they call cytopathic effects. As you can see, there is some changes in the morphology. There are these plaques that form, and this is a sign of, the cells are in the dying process. And this is what's put forth as the proof that there was a virus way back in the original sample of the lung fluid, even though it's not known what else was in that lung fluid. They sometimes do some molecular testing to rule out certain known bacteria and viruses, but there are flaws because many of those tests are not validated. But they really don't do a compositional analysis and say what's everything that's in there, and of course there's no control experiment. So it's really erroneous to interpret it this way. So back to the issue of control experiments. Were any control experiments ever performed? So this is a paper by John Enders, and this is the original paper where this method was developed, the tissue culture isolation experiments. And Enders won the Nobel Prize related to the polio vaccine for developing this procedure, but it was developed as a manufacturing procedure of vaccines. So they used to think you could only, quote unquote, culture viruses using the type of cells that it infects. So for polio, using spinal cord or nerve cells. As some of you may know, those are very difficult to culture in a laboratory. So he had the idea to use some kind of fetal cells, which were much easier to grow. They're, practically immortal in the laboratory. And he mixed in, the toxic ground up spinal cord from people who died of polio in with the fetal cell culture, and was able to use that to manufacture the polio vaccine. And I think he helped either Salk or Sabin, I don't remember for sure. But nonetheless, in this paper, which is about measles, he used the same procedure with a different cell line, but it wasn't necessarily for the purpose of manufacturing a vaccine. In this case, or at least it wasn't published, it was for investigation. They talked about propagation in the tissue cultures. But in this experiment where they did, essentially what I just described, except they added a few other things like milk to the sample, they did the procedure without a source of measles and got the same exact results, the cytopathic effects. And they, wrote in here that that could either be from another virus from the kidney cells, unknown, of course, always blame a virus, or it could be from unknown factors. right? But never considered that it was the tissue culture conditions themselves that could have been the cause, even though that is the purpose of doing a control experiment. And in the conclusion of this paper, by the way, it doesn't say that it proves the existence of a virus, the cytopathic effects. And it also says specifically that what they found in vitro does not necessarily correlate with anything that happens in the patient with measles. It was only after this publication and after the Nobel Prize was awarded that virologists began adapting this procedure suddenly to prove the existence of viruses rather than to study viruses or manufacture vaccines.
In plain English, alleged viruses should break down the cells from healthy human adults in a test tube as well. But since their experiments failed to do any such thing, they switched to more susceptible, chromosome unstable kidney tissue from green monkeys, or alveolar A549 cells from human lung cells sick and dying with cancer, already breaking down naturally by cytopathic effect instead. Dr. Charles Dauger, the electron microscopist for Dr. Luc Montagnier, credited for discovering the alleged HIV virus particle, said, quote, "We have never seen HIV virus particles in the purified virus gradient. What we have seen all the time was cellular debris, or the aftermath of already broken down cells, not HIV virus particles." It doesn't matter how cellular tissue dies, whether you starve and poison it via necrosis, or it dies naturally from apoptosis, or you just squish it to death between two glass plates like this. When you look at the aftermath under an electron microscope, you get this dead cellular debris that looks like what are alleged to be virus particles. The ones on the left, made by squishing a cell to death between two glass plates. The ones on the right, made by virologists starving and poisoning a cell to death in a test tube or petri dish, and then poisoning and destroying it even further to view under an electron microscope. Or as they say in Thailand, same but different. In a June 2020 paper entitled Severe Acute Respiratory Syndrome Coronavirus 2 from Patient with Coronavirus Disease, United States, when this group attempted to culture SARS-CoV-2, they had no desired result with human adenocarcinoma cells, A549, meaning human lung cancer cells, no luck with human liver cells, HUH 7.0, or with human embryonic kidney cells, HEK-293T, or even with a big brown bat kidney cell line, EFK3B, but then miraculously declared they had a viral isolate following the observation of cytopathic effects in green monkey Vero E6 kidney cells. They admitted from the wells in which cytopathic effect, meaning the killing, breakdown, and deterioration of cells into small particles were observed, meaning after adding the exact same lung sputum sample from a single patient alleged to have SARS-CoV-2 to 5 different types of cell tissue lines, then starving and poisoning those tissues, they still only found that procedure only killed the cells of green monkey kidney tissue and not the rest. Hey, 1 out of 5 ain't bad. Of course, if you only had a 1 out of 5 chance of your parachute opening, would you trust the parachute science enough to jump out of an airplane? As is typical, there seemed to be no sense of irony for them that the purported human respiratory virus cannot be shown to infect the relevant cell type, let alone the relevant human species, and their experiments were once again invalidated by the absence of appropriate control cultures. Why not at least use African green monkey lung cells instead of kidney cells, if this is an alleged respiratory virus and not a kidney virus? And if you're not an African green monkey yourself, and even their own experiments can't break down decrepit alveolar A549 human lung cells already dying of cancer in a test tube, why would you believe your own healthy lung cells in your own nutrient-dense body would be more susceptible to such an alleged but unproven theoretical virus? Nonsense. And since they refuse to scientifically control their own cell culture experiments and debunk themselves, they instead control our societal culture itself by changing the societally agreed language definition of the word isolation to propagate their germ theory fraud. And that's why isolation matters. A further and major embarrassment for virology is that alleged viral particles that have been successfully purified after an unpurified cell culture beginning have not been shown to be replication competent, meaning able to multiply in a new host cell, or disease-causing by themselves. In other words, what particles have been at least partially physically quote-unquote isolated, along with the other junk in the test tube, never directly from a living being as they should be found, but only after tissue culture contamination, can only be said to be extracellular vesicles. Nothing more, nothing less. In May 2020, a publication appeared in the journal Viruses that claimed, nowadays, it is an almost impossible mission to separate extracellular vesicles and viruses from each other by means of canonical vesicle isolation methods, such as differential ultracentrifugation, because they are frequently co-pelleted together due to their similar dimension. Meaning, because the particles they allege to be viruses are the exact same size, weight, shape, and density as all of the other identical cell debris particles, they say after they centrifuge them that it's impossible to take out the alleged virus particles only with a pipette, because they are in the exact same density band as all of the other junk, just like this cherry tomato is in the same layer as the dish soap. They are admitting they can't suck out the cherry tomato without the dish soap coming with it, or vice versa. Nowadays means in contrast to how they've always done this since the beginning, and it is unclear how such an observed technical change may be reconciled with biological laws. They've just now figured this obvious fact out for the first time in 60 years. It appears more likely that the virologists are distancing themselves from their own techniques in order to avoid refutation of their own false postulates. They may have to accept that the reason it is impossible to separate extracellular vesicles from viruses using differential ultracentrifugation is because their assertion that viruses are present in the sample are ill-founded, meaning their alleged viruses are extracellular garbage debris, nothing special or different. In 2017, the Perth Group pointed out in their magnum opus, HIV, a virus like no other, that in virology, while purification retains its everyday meaning, isolation is an expediential term, meaning advantageous to them, that virologists assign to data, not physical reality, they claim are proof a particular virus exists. The paper states neither virologists nor the scientific literature provides a satisfactory definition of virus isolation. HIV expert Jay Levy defines virus isolation as a sample of a virus from a defined source. White and Fenner as the ability to identify a totally unforeseen virus or even discover an entirely new agent. Montagnier and Weiss as propagating them, viruses, in a cell culture. The 2013 6th edition of Field's Virology defines virus isolation as Viruses can be isolated from an infected host by harvesting excreted or secreted material, blood or tissue, and testing for induction of the original symptoms in the identical host, or induction of some abnormal pathology in a substitute host or in a cell culture. Once the presence of a virus has been established, it is often desirable to prepare a genetically pure clone. It goes without saying that if virus isolation is to take a sample of a virus from a defined source, or propagating viruses in cells in culture, one must first have proof that the virus exists in a defined source or in cells in culture. Neither is virus isolation induction of some abnormal pathology, or once the presence of a virus has been established.
There's no universally accepted definition for these terms, strain, variant, and isolate, in the virology community. So how can we expect that everyone else gets them right? What happens is you're trained in someone's laboratory and you hear them say things and you associate a meaning with them, and that's what you do. And they may or may not be right. Now, despite this looseness of definitions, for example, there's no one place where you could go and find the definition for variant, for example, or isolate, or strain. I looked in several textbooks. In fact, not even my textbook, Principles of Virology, co-authored with 4 other virologists. Not even there will you find a definition of these terms. I think we need to put it in the next edition, though. Let's go through and try and define them and put them in some kind of context with the current outbreak. Let's start with isolate. What is an isolate? An isolate is a virus that we have isolated from an infected host, and we have propagated that in culture.
You cannot define the word isolate with the word isolation or isolated as its definition or meaning. Defining something with its own name is called recursion, known as a homunculus logical fallacy. Just like you do not define the word red as something that is red in color. A blind person or colorblind person would have no idea what you are talking about, or a child who hasn't learned the names of colors yet. So you say, red is a color whose hue resembles that of blood or of the ruby, or is that of the long wave extreme of the visible electromagnetic spectrum. To a child, you point to the red blocks only and say that is red, not those other colors. You do not point to a starved and poisoned tissue culture with tens of millions of pieces of broken down genetic material from a dozen different sources and say that is an isolated virus, any more than you would think this is a pile of isolated red blocks. Virology invented the hypothesis or theory of viruses, so whatever method it employs in an attempt to prove their existence, it must satisfy that definition. At the heart of the matter is a simple concept, and we need to see evidence that alleged disease-causing particles cause new particles that are clones of the former. Claiming that detected proteins and nucleic acids are of a specific viral origin and not just normal broken down components of a tissue culture is not possible unless the alleged viral particles have been truly isolated by purification and shown to have these key biological characteristics. As outlined by the Perth Group in HIV, a virus like no other, purification is necessary to prove the existence of viruses for several reasons, including the following. Viruses allegedly replicate only in living cells. Since cells and viruses are composed of the same biochemical constituents, separation of particles from cellular material is essential for defining which nucleic acid and proteins belong to the virus particles. Also, to prove the particles are infectious. In other words, it is particles, not other factors, that are responsible for the production of new particles. This requires purification of both sets of particles. also to demonstrate their biological and pathological effects, and to obtain antigens, proteins, and nucleic acids for use in antibody and genomic tests, respectively.
Like you were alluding to, everything's, flipped the other way with Snopes, right? It's the same thing here. If you actually go to the studies and the information, the way in which they do the lab information, the studies, and they gather their data and they make those claims is all based on artifacts that they're manufacturing in the laboratory process, and is not what happens in the physical body, in the living body. And this has been the major con that they've done to our medicine and to the knowledge base, is that they're not watching and looking at the living blood and how things operate. They are always removing particles, removing tissues, altering them through their methods, whether it's petri dishes, antibiotics, other types of cells like kidney cells from the monkey, or fetal cells, or their methods of growing the so-called virus. And then they're adding, coloration and adding UV light and other light when they're, viewing things under microscopes. And they're actually manufacturing an illusion with artifact production. And now they're taking artifacts and trying to study them further and make claims about them. To me, it's absolute insanity, because they've never done the control studies and the proper methods to then corroborate it back with the story that happens in the living body. They're assuming that what happens in the petri dish or under the microscope is what happens inside of us. That is a huge glaring error of science. It's not scientific method structured, and it's not validated. They don't ever validate those things.
You've probably heard the term virion, meaning a theoretical complete viral particle consisting of RNA or DNA surrounded by a protein shell and constituting the infective form of a virus, though no proof of one exists other than a wild guess as to what these destroyed tissue culture particle images may possibly be made of or possibly do. They theorize them to be possibly like this, just like a pink-horned unicorn horse theoretically might look something like this, or maybe even something like this. Although less common, virologists will sometimes obfuscate the meaning of purification as well. Purification means the removal of contaminants from something, meaning getting rid of everything else in the tissue culture and leaving only the alleged virus particles, or the process of extracting something from a substance, meaning taking out the alleged virus particles only and leaving all the other tissue culture garbage there. On May 23, 2022, Belgian professor of virology Mark van Ranst claimed that with regards to SARS-CoV-2, in another article, they have further purified the virus by ultracentrifugation in beta-cyclodextrin. Van Ranst was referring to a 2008 paper that described large-scale preparation of UV inactivated SARS coronavirus virions, which related to the purported SARS-CoV-1 virus. However, this paper simply outlines a protocol claiming to purify virions, and there is no part of the paper that demonstrated the existence of any replication-competent particle. All that was shown were some low-quality images purporting to show infected Viro E6 cells. With regards to the check of purified virions following centrifugation, no images were provided, but the claim was made anyway that the concentration of purified virions is determined by BCA or bisynchroninic acid assay with bovine serum albumin as a standard, which is a joke because all a BCA or bisynchroninic acid assay can do is detect the total concentration of a protein in a solution. It cannot determine whether that protein is from a virion or just plain old regular proteins abundant in tissue cultures. In other words, It can tell you there are exactly 5 hoofed, 4-legged animals with tails and manes here, but not if they're unicorns or just regular horses. In an October 2013 paper in the journal Nature, we find a paper entitled Isolation and Characterization of a Bat SARS-like Coronavirus that uses the ACE2 receptor. We are given this ridiculous image of completely different vesicle particles in size, shape, and color. If I were an alleged scientific expert on cloning, what would you think if I claimed that these people were all replication-competent identical clones of each other? Yet, the convenient variation in particle size is apparently because coronaviruses usually have a diameter, excluding projections, of between 80 and 120 nanometers, although in extreme cases, the diameter can vary between 60 and 220 nanometers. Because the definition of identical clones is clearly as loose as the definition of isolation to virologists. But you see, Mark Van Rans cites a fancy-worded fluff paper, sodium dodecyl sulfate, polyacrylamide gel electrophoresis of proteins, which sounds so scientific that you'll never bother to question it. By really just claiming that confirming the existence of a virion or virus particle using gel electrophoresis, which just simply separates different proteins from each other by their molecular mass, is a better way, is as big of a joke as using bisynchronicic acid assay, because gel electrophoresis too cannot provide any evidence that a protein even belongs to an alleged virus or so-called virion. Only that it is a different mass and density weighted protein than other proteins with different molecular masses. But at least his way can tell a big yellow horse from a little brown horse, but still not if it's a unique yellow unicorn or just a plain old yellow horse. But Mark Van Ranst is a virology expert, so we should trust him, right?
To tell you about my experiences being the crisis manager, the flu commissioner for Belgium, and highlighting the communication aspects there. And then you have one opportunity to do it right. I mean, day one is so important. In day one, you start your communication with the press, with the people, and you have to do it right. I mean, you have to go for one voice, one message. In Belgium, they chose to appoint a non-politician to do that. I mean, I have no party affiliations, and that makes things a little bit, at that time at least, a little bit easier, because you're not attacked politically, majority, minority. That doesn't come into play, and that was a huge advantage. The second advantage is that you can play in Brussels the complete naive guy and get a lot more done than you would otherwise be able to do. You have to be omnipresent that first day or the first days, so that you attract the media attention. You make an agreement with them that you will tell them all, and if they call, you will pick up the phone. When you do that, then you can profit from these early days to get complete carpet coverage of the field, and they're not going to search for alternative voices there. And if you do that, that makes things a lot easier. And then you have to say, okay, well, we will have H1N1 deaths. Of course, that would be unavoidable. I used there Sir Donaldson's quote, where he said that in the UK, by the peak of the epidemic, 40 people would die per day at the end of the summer. So 62 at that time million people in the UK, 40 deaths a day. I worked it out for Belgium. That would be 7 deaths a day at the peak of the epidemic. I used that in the media. 7 Belgian flu deaths per day at the peak of the epidemic would be realistic. That is true in every year, even interpandemically. That is very, very conservative. However, talking about fatalities is important, because when you say that, people say, wow, what do you mean? People die because of influenza? That is true in every year, even interpandemically. That is very, very conservative. However, talking about fatalities is important because when you say that, people say, wow, what do you mean? People die because of influenza? And that was a necessary step to take. And then, of course, a couple of days later, you had the first H1N1 death in the country, and the scene was set and it was already talked about.
Van Ranst was not the only virologist making claims about purifying viruses. In response to an email inquiry, Dr. Marissa Grossages from the Robert Koch Institute responded that, "We purified SARS particles by density gradient. However, just, meaning only, from the cell culture-derived virus, as you wrote. The challenge with purifying SARS from patient samples is that you won't get a visible band." Meaning, according to her, if you take the sputum from a person who is allegedly sick from millions or billions of SARS replicating viruses in their lungs, and hence in their lung sputum, and centrifuge that, they can't find a density band consisting of any of these alleged SARS virions anywhere in the test tube. But they can magically find particles they claim to be SARS virions after their tissue cultures are starved and poisoned and break down into a million pieces, or should we say rather, conveniently find them. When pressed for further details about how or if the experiments were even controlled, she responded, "We are not allowed to share any protocols with a private person. I can only refer to our publications, where infection experiments are described in detail." Unfortunately, no such promised details were given in any of the experimental studies she provided. SARS-CoV-2 remains nothing more than a hypothetical computer construct assembled from genetic fragments of unproven provenance. It's as if outside of a chemically controlled and manufactured petri dish, viruses are nothing more than a figment of the imagination. As Mark Bailey points out, there has never been a physically isolated, i.e. purified particle, shown to be responsible for the production of identical particles or a particle shown to be the cause of pathological effects in any human or in an experimental animal model. In other words, they've never observed any submicroscopic particles they label viruses do any of the things they claim they do. Thus, the declaration by virologists such as Mark Van Ranst, along with the WHO and its adherents, that an infectious particle termed SARS-CoV-2 is causing a disease pandemic is shown to be patent scientific and intellectual fraud. So what is virology? When startled, the mythical Uzlam bird will take off and fly around in ever-decreasing circles until it somehow manages to fly up its own ass, disappearing completely, which adds to its rarity. It is hard to know exactly what to call virology, but it is not science. The current practitioners are engaging in some form of algorithmic or statistical speculation added to circular reasoning and confirmation bias, searching for, interpreting, and recalling information in a way that supports their theory, or what we commonly call cherry-picking data. With a complete absence of what should be the corresponding process of refutation that lies at the heart of the scientific method, i.e., trying to debunk their own theory with valid control experiments in the lab and actual observations of isolated particles causing contagion and illness in the real world, not just cartoons and Hollywood movies. While the abandonment of the scientific method may be accidental or unnoticed by lower-level virologists and lab technicians, there are almost certainly conspiratorial motivations at higher levels of the globalist hierarchy. For example, the WHO, the Center for Disease Control, and the United Kingdom's Health Security Agency are all parties to virology's deceptive practices. However, the anti-scientific practices are also replicated in most other countries, whether this relates to claims of virus isolation and the wholesale misapplication of the polymerase chain reaction, or PCR test, for clinical diagnostics, or a failure to disclose the crucial control details involved in virus culture and genome creation. Pseudoscience consists of statements, beliefs, or practices that claim to be both scientific and factual, but are incompatible with the scientific method. In other words, pseudoscience cannot be proven, tested, or repeated. It's just a baseless theory, like the pseudoscience germ theory of disease. Science, and even solid scientific theory, can be proven, tested, repeated, and most importantly, falsified, meaning solid scientific theory can be demonstrated to be false. You can have a pseudoscientific theory, like flying unicorns actually fly backwards instead of forwards, but it's impossible to either disprove or falsify that theory or prove that theory true, because there's no such thing as flying unicorns in the first place. Refutation is at the heart of genuinely testing a legitimate scientific theory through the scientific method. As Karl Popper wrote in his book, The Open Society and Its Enemies, about scientific theory and method, so it is, I hold, the possibility of overthrowing it, the scientific theory, or its falsifiability, that constitutes the possibility of testing it, and therefore the scientific character of a theory, and the fact that all tests of a theory are attempted falsifications of predictions derived with its help, furnishes the clue to the scientific method. The view of the scientific method is corroborated by the history of science, which shows that scientific theories are often overthrown by experiments, and that the overthrow of theories is indeed the vehicle of real scientific process. The contention that science is circular cannot be upheld. But for science to be true and not just pseudoscience, it must be logical and reasonable. Yet circular reasoning is a logical fallacy, meaning illogical and unreasonable, in which the reasoner begins with what they are trying to end with. The components of a circular argument often seem logically valid, which is why the pseudo-virologists use circular reasoning, because if the premises are true, the conclusion must be true. But a premise simply means an assumption or theory. As Karl Popper said, your control experiments are there to try and debunk your own premise, assumption, theory. Real science does not fix and rig experiments through dodgy, unscientific methods to make their baseless claims appear to be true, and then not bother to run any control experiments to debunk or falsify their own premise. But I'll tell you who does do that: con men. This woman is sick with flu symptoms. I wonder if there are submicroscopic particles called viruses inside of her, causing these symptoms. Virologists will tell us. Step 1. Collect her lung sputum containing the virus particles. That is circular reasoning. You are supposed to try and disprove through falsification experiments your premise that those particles are even there in the first place, not baselessly assume for no reason whatsoever other than your virology school-taught belief that they are there before you even start. Circular reasoning is illogical, unreasonable, and thus unscientific. Dr. Bailey was correct when he stated, "It is thus a reasonable question to ask. Has virology ever been a scientific pursuit?" With regard to the scientific method, the virologists create unfalsifiable hypotheses by setting up paradigms where any number of observations, whether it be illness or alleged test results, can be attributed to their viruses. The observations are passed off as proof of virus existence in the manner of a circular loop of reasoning that no longer requires the demonstrable existence of a virus. Any claims of reproducibility, for example, in the form of a PCR process or purported viral genome, are simply more circuits of the same loop. Historically, virology has been characterized by a lack of valid control experiments, and none of its foundational claims have been established through proper exercise of the scientific method. Again, falsifiability. They should take sputum from a completely healthy person and assume there is no virus. At least make sure that person tests virus negative with the PCR test, even though it doesn't matter. Then poison and starve that healthy sputum on the exact same cell culture using the exact same chemicals for the exact same period of time. Let it all break down into 56 million tiny genetic fragments, and then tell their computer to arrange those fragments like a puzzle until they match the fictional computer in silico genome for that alleged virus. If they can do that, which virologist Dr. Stefan Lanka did successfully do himself, it proves it is their procedure causing the cytopathic effect breakdown of tissue in their culture and not an alleged virus. The first alleged virus to be discovered was the tobacco mosaic virus, which looks remarkably just like rotting lettuce in your cold fridge. And one of the proofs for this is said to be contained in Dmitry Ivanovsky's 1903 treatise about the mosaic disease of the tobacco plant. However, it is patently clear that Ivanovsky's described experiments lack any valid control comparisons and were thus, surprise, surprise, unscientific and inconclusive.
Here we've got the conflation of sick plants with a virus, where we're jumping from A to Z here without any of the proof that's required in the middle. So one thing, we got sick of hearing this too, that Ivanovsky is the father of modern virology, and he's got this great paper, late 1800s, 1900, we're talking now, and that this proves that this was the first ever documentation of a virus. But if you read that paper, Ivanovsky doesn't claim that he's found a virus in the modern conception, and I think that's where the public get confused, and even people who are doctors and scientists in the system get confused as well, because they don't actually read the source material. Ivanovsky simply observed that some plants got sick and others didn't. He even notes that it seems to be related to where they're planted, i.e. the terrain, the environment. And we all know that you can't plant lemon trees in Greenland. We don't say that, goodness, we don't say every single citrus tree we've planted in Greenland has come down with some sort of virus, because it just won't take, So if you actually read Ivanovsky's paper, he talks about this contagious fluid that he's run through filters. But again, the methodologies he used were insufficient, because he's simply taking material from one sick plant and injecting it into a healthy one and watching it get sick and saying, well, there must be some sort of agent, transmissible agent. And yes, there is an agent, but it's not contagious or transmissible, because grounding it up and injecting it into another plant doesn't replicate what happens in nature. So Ivanovsky, there's nothing there. And it's clear, he even says in those experiments that he planted a sick plant, which was alleged to have the tobacco mosaic virus, but we should just say it's got tobacco mosaic disease is more accurate. And he planted healthy plants right next to it, within centimeters. And hey presto, that plant didn't get sick.
Yeah, so where's the virus?
Yeah, his own experiments basically document that it didn't transmit, and unless he did these abnormal kind of grind up and inject methods, which went completely out of control.
It is tempting to forgive the early pioneers of virology that their uncontrolled and unscientific methodologies were simply typical practices for that era. However, Germ theory critic Claude Bernard offered the following insight into the importance of control experiments when adhering to the scientific method decades earlier in 1865. If indeed we characterize experiment by a variation or disturbance brought into a phenomenon, it is only insofar as we imply that the disturbance must be compared with the normal state. As experiments indeed are only judgments, they necessarily require comparison between two things, and the intentional or active element in an experiment is really the comparison which the mind intends to make. Bernard was advising the need to have a valid control, or some suitable comparison, to ensure it was only the new experimental element that was causing an outcome. In plain English, if you suspect a dress makes you look a bit fat, you need to try on a bunch of other dresses to see if you look skinny in those, to make sure it's not just the fat underneath the dress that makes you look fat. It is possible Bernard was the only scientist of his time in virology's pursuit of invisible enemies who actually understood the scientific method, but not probable. Moving forward to another early claimed virus discovery, the textbook Retroviruses informs us that, in 1911, Peyton Rouse at the Rockefeller Institute in New York reported the cell-free transmission of a sarcoma, meaning a tumor arising from the connective tissue, in chickens. The virus, isolated by Rouse, bears the name of its discoverer, Rouse sarcoma virus. However, a review of Rouse's paper, A Sarcoma of the Fowl, transmissible by an agent separable from the tumor cells, reveals that he did not claim to isolate anything, let alone anything that met the definition of a virus. His methodology involved grinding up chicken tumor material, filtering it, and injecting it directly into other chickens, with the observation that some of them would also develop tumors. He reported that the control experiments consisted of injecting unfiltered tumor material into chickens, which tended to result in much larger tumors. Rouse postulated, meaning took a wild guess, at the presence of a causative ultramicroscopic organism, but conceded that an agency of another sort is not out of the question. Indeed, the experiment failed to provide any evidence of an infectious and replicating particle. It simply showed that diseased tissue introduced by an unnatural and invasive route into another animal could cause it to exhibit a similar disease process. Would you be shocked if someone ground up this rotting sarcoma tumor, injected it into your connective tissue, and you developed a tumor at the injection site? Would you say you caught a sarcoma virus there? This debunks William Guy's 1925 work, because finding a tumor is not proof that a virus caused the tumor. It simply demonstrated tumor formation following injection of diseased tissue. Further, it was determined in 1927 that sarcoma of the fowl could be induced by the injection of dilute arsenious acid in foreign embryonic pulp. The carcinogenic effects were also replicated following the equivalent bacteriological filtration that Rouse performed, and the disease was shown to arise from the foreign tissue, not from the host tissues. Carrel's summary stated, "We have discovered the presence of an important phenomenon, the formation by the tissue under the influence of a nonspecific chemical substance of a principle which resembles a virus, acts in a specific fashion on the cells of a certain type, and reproduces itself indefinitely in the presence of these cells." The viral hypothesis should have been thrown out right there, but instead, half a century later, the establishment kept it alive and rewarded Rouse with a Nobel Prize in 1966 for his discovery of tumor-inducing viruses. Then in 1954, when John Enders and Thomas Peebles claimed they had propagated the measles virus in human and monkey kidney cells, no further tolerance should have been extended to virology's unscientific experiments. Enders and Peebles added throat washings and blood to their cell cultures, and on observing cytopathic effects, or the dying and breaking down of cells in their test tubes, concluded that the in vitro or in scientific test tube appearances might be associated with the virus of measles. Might is used to indicate a condition or state contrary to fact, or used to indicate a possibility or probability that is even weaker than may. They did at least warn that cytopathic effects, which superficially resemble those resulting from infection by the measles agents, may possibly be induced by other viral agents present in the monkey kidney tissue itself or by unknown factors, but then went on to inappropriately conclude that this group of agents is composed of representatives of the viral species responsible for measles. They did do one lazy control experiment where they admitted a second agent was obtained from an uninoculated culture of monkey kidney cells, meaning they didn't put any material from a person with measles in there, and that the cytopathic changes it induced in the unstained preparations could not be distinguished with confidence from the viruses isolated from measles. That actually invalidated the evidence for their conclusion, but ideally, several control experiments should have been done, some with no human-derived samples added, some with human-derived samples added but from people who are well, and some with human-derived samples from unwell subjects but said not to have measles clinically or some other alleged viral condition, just sick people. That is how you do real science, and this is how you do not. The virologists, however, have continued to repeat the uncontrolled methodology of Enders and to this day claim that such cytopathic effects are incontestable evidence of viruses. Dr. Stefan Lanka has documented the history of these unscientific practices and in 2021 demonstrated that cytopathic effects could be induced in cell cultures by the laboratory process itself. The results of Lanka's experiments are depicted here, proving you can easily and repeatedly break down healthy tissue in a lab causing cytopathic effect without adding any material sample from a sick person, simply by following the cell culture procedure techniques virologists adopted from Enders and Peebles. In many virology publications, a control or mock-infected experiment is mentioned, but the details of such experiments are conspicuous by their absence. Much like our mock unicorn control here. A Northwestern University, Illinois webpage states that mock-infected means a control used in infection experiments. 2 specimens are used, one that is infected with the virus vector of an interest, and the other is treated the same way except without the virus. The definition is already flawed, as terms such as virus and infected have been introduced and thus presumed to exist before actually being established. In June 2022, in response to an Official Information Act request concerning the paper, characterization of the first SARS-CoV-2 isolates from Aotearoa, New Zealand, as part of a rapid response to the COVID-19 pandemic, the University of Otago stated, the paper published by Professor Quinones Mateu and colleagues was a descriptive paper. This means there was no hypothesis to prove or disprove. In 2008, the journal Infection and Immunity featured a guest commentary titled Descriptive Science that explained why descriptive research by itself is seldom conclusive and may simply serve as a starting point to orientate further investigations. The authors pointed out that microbiology and immunology are now experimental sciences, and consequently investigators can go beyond simply describing observations to formulate hypotheses and then perform experiments to validate or refute them. As this essay outlines, the virology establishment will not divulge or carry out these required experiments, seemingly in order not to refute itself. It intentionally limits itself to ongoing opportunistic fishing expeditions backed by confirmation bias, thus disqualifying itself from the scientific method due to its inconsistency with the hypothesis-driven and falsifiable approach described earlier by Popper. If we're supposed to trust the science, should they not at least be beholden to actually use the scientific method and not hypothetical descriptive science? Dr. Mark Bailey has previously written in a postscript derived from A.F. Chalmers' book, What Is This Thing Called Science?, that one of the pivotal issues with virology was that it invented itself as a field before establishing if viruses actually existed. Literally the cart coming before the horse, and it has been trying to justify itself since its inception. But if pink unicorns have never been proven to exist, why invent unicornology? And don't you think unicornologists might have some incentive to cheat to prove they do exist in order to keep those big paychecks coming in? As Dr. Mark Bailey wrote in Warning Signs You've Been Tricked by Virologists, in this instance, a virus particle was not observed first and subsequently viral theory and pathology developed. Scientists of the mid and late 19th century were preoccupied with the identification of imagined contagious pathogenic entities. The observations of the naive inductionist did not identify a virus a priori and then set about studying its properties and characteristics. The extant presupposition, meaning just conjecture or presumption, of the time was that a very small germ particle existed that may explain contagion. What came thereafter arose to fulfill the presuppositional premise. We shouldn't even call it the germ theory of disease, because even a scientific theory demands evidence that has repeatedly been tested and corroborated in accordance with the scientific method. And so it is clear that viruses have never even reached the stage of a theory. According to the science itself, they remain mere speculation. And thus, virology's lack of controls means it is not a scientific pursuit. OIA requests have revealed that New Zealand's Institute of Environmental Science and Research, who have claimed isolation and genomic sequencing of the SARS-CoV-2 particle in the Antipodes, are also guilty of failing to perform any valid controls. In the tradition of Enders, they have not paused to check whether the cytopathic effects they witnessed or genomes they assembled via computer simulations could also be created in valid control comparisons. That is, by performing experiments with other human-derived specimens from both well subjects and unwell subjects who are said not to have the alleged disease COVID-19. Instead, ESR described their insufficient negative control in which the flask undergoes the same conditions as the flask used for viral culture. However, we use infection media only. Again, the whole point of control experiments is to test whether an independent variable can be identified, not change multiple variables and assume that the results of the experiment can only be explained by one thing, in this case, an imagined virus. The central conductor in these anti-scientific pursuits is the World Health Organization. It is very telling that in their 94-page genomic sequencing of SARS-CoV-2 document, there is a mere 4 sentences discussing control samples. However, neither of these negative or positive controls are sufficient to validate the genomes that the virologists are producing through these techniques, because they can only serve to calibrate the pipeline. And as become apparent, the WHO cannot point to one valid positive control experiment. Yet, on February 11, 2020, they named the new disease they had invented, COVID-19, with the associated claim that it was caused by a novel coronavirus. They have provided the green light for anyone around the world to find SARS-CoV-2 in their backyards without the need for valid control experiments. Yet, there is a clear necessity for comparative controls where similar patient sputum samples without the alleged virus, like these two healthy and sick people who tested negative, are processed in the exact same way, so that only one variable is being tested. Comparing the results of a sputum sample alleged to contain the virus from a positive PCR or antigen test with one of the negative controls described by the WHO's document above cannot validate the process, as the latter samples of buffer or water do not contain the sputum genetic muck soup that is part of the former sample. With all of the failures to culture postulated viruses, modern virology now favors direct metagenomics of crude samples. The NIH tells us metagenomics is the study of the structure and function of entire nucleotide sequences isolated and analyzed from all the organisms in a bulk sample. That is not isolation. We're no longer interested if there really are virus particles in this tissue culture soup, or if there really are green blocks in this pile, because we know blue and yellow mixed together make green, and there are blue and yellow blocks in this bulk sample. So if our computer Castle block genome template requires green blocks that aren't in the sample, we'll just sequence all the blue and yellow blocks in this pile together and say this bulk sample technically has green blocks in it where the blue and yellow primers meet. Could you make this castle out of these blocks? Virologists can. That's exactly how metagenomic computer software can assemble 56 million random short 150 long ACGT nucleotide sequences from a tissue culture rich with multiple broken down genetic sources into any 30,000 sequence long genome you tell it to, be it an alleged virus genome or a banana genome. And that doesn't even include the probability algorithms programmed into the software when it can't fit certain pieces together precisely and needs to take a wild guess, a fictional computer-created genomic band-aid, if you will. Is there any recipe you couldn't make if you had 56 million ingredients at your disposal? They sometimes call this shotgun sequencing, and subsequent artificial computer assembly of these genetic fragments allows virologists to create new in silico viruses, meaning viruses existing in a fictional computer model construct only, out of thin air. This software invention then provides other virus hunters with pre-designed PCR primer panels, so they can also discover the same meaningless sequences and claim it is the same virus. The artist no longer has to manually mix yellow and blue to make green. The computer's software just assumes green is there if it detects any yellow and blue, and then puts green in the sequence automatically, whether it is really there or not. Like claiming the Ukrainian flag is technically green, if you grind it up into 56 million micro pieces and restitch it all together in alternating micro colors. ESR were involved in a publication in which they proclaimed the discovery of SARS-CoV-2 in 9 subjects through this methodology. But when asked to provide all details of the control group that was used when comparing the results of sequencing, they refused and instead sent links to SARS-CoV-2 sequencing protocols. If ESR were using these protocols, then we can see that they are endorsing insufficient controls that are described as a negative control of nuclease-free water, while an optional positive control can also be included, which may be a synthetic RNA constructs or high-titer clinical sample which can be diluted. Once again, these types of controls can only serve as pipeline calibration techniques, not the validation or the clinical significance of any genomes they assemble. On July 19th, in response to an OIA request, they stated that ESR has not performed any experiments to scientifically prove the existence of SARS-CoV-2 virus and can therefore not provide you with any records. On the 17th of August 2022, in response to another request, they admitted that ESR has not performed any experiments to scientifically prove that the SARS-CoV-2 virus causes COVID-19 and can therefore not provide you with any records. Nobody else has performed these required scientific experiments either. So they have to rely on animal abuse and antibody studies. With the inability to demonstrate the physical isolation of a disease-causing particle that meets the definition of a virus, the virologists have engaged in animal experiments to convince the uninitiated that such pathogenic particles exist. And like clueless monkeys studying other monkeys, these animal experiments also lack valid control experiments. An illustrative example of this monkey business was the paper Comparative Pathogenesis of COVID-19, MERS, and SARS in a Non-Human Primate Model, published in May 2020 by a team that included none other than Christian Drosten and Ron Fauchier. The nonsense of what was published in Science can be summarized as follows. The 8 cynomolgus macaque monkeys in the experiments were inoculated with SARS-CoV-2 under anesthesia via a combination of intratracheal 4.5 milliliters and intranasal 0.25 milliliters per nostril routes. Pouring fluids down the nose and throat of a sleeping monkey is not a natural exposure route, and 4.5 milliliters poured into a small 3 to 5 kilo monkey's lungs is equivalent to pouring around 1/3 cup of foreign biological material into a human's lungs while they're asleep. The inoculum poured into their lungs and nose was made from SARS-CoV-2 isolate beta-CoV Munich BAVPAT1 2020, obtained from a clinical case in Germany from a German traveler returning from China. The virus was propagated on Viro E6 cells, unstable green monkey kidney tissue cells, in Optimum, Glutamax, and then supplemented with kidney nephrotoxins, penicillin, and streptomycin, not to mention the kidney-dissolving enzyme trypsin. They have asserted that they have a viral isolate, yet neither they nor their supplier have demonstrated the existence of a virus in the sample. All that can be said is that the sample contains foreign biological material from the human-derived clinical sample and monkey kidney cells, in addition to cellular breakdown products and 2 antibiotics. Even after being waterboarded in the nose and lungs with this noxious fluid, none of the older macaques showed the severe symptoms that humans do. In fact, no overt clinical signs were observed in any of the infected animals, except for a thin serous nasal discharge in one aged animal on day 14 post-inoculation. No significant weight loss was observed in any of the animals during the study. In other words, despite the direct entry into the lungs with what they claimed was the SARS-CoV-2 virus, it didn't make any of the monkeys noticeably sick. But yet it allegedly killed nearly 7 million people? By day 14 post-inoculation, or being waterboarded with toxic muck, all remaining animals seroconverted, meaning they developed antibodies in their blood serum, allegedly as a result of infection or immunization, as revealed by the presence of SARS-CoV-2 specific antibodies against the virus S1 domain and nucleocapsid proteins in the sera. Yet these S1 and nucleocapsid proteins have never been shown to be viral in origin, regardless of whether they induce the detection of other proteins termed antibodies in a host. The virologists once again employ circular reasoning to claim that the detection of an antibody proves the existence of a virus because the antibody is declared to be specific to only the alleged virus. Despite antibody scientists themselves assuring us that these so-called antibody proteins bind to all sorts of non-specific things or nothing at all, depending on the environment, the state of the proteins, whether acidic or basic, oxidized or reduced. Every scientist who has carried out such experiments or studied them knows this, yet every virologist conveniently seems blindly or willfully ignorant of this simple fact. In fact, we see this same circular reasoning with vaccines. How do I know the COVID-19 vaccine works? We can reliably prove it with an antibody test. Yeah, but an antibody test confirmed that I'm already immune to COVID-19. You can't reliably prove that with an antibody test. As a measure of so-called virus shedding, nasal, throat, and sticking Q-tips up the rectum of the monkeys were analyzed for virus by reverse transcription quantitative polymerase chain reaction, or RT-PCR. But there was no virus shedding. There was simply detection of the same sequences that had been recently poured into the monkey's respiratory tracts. It's like pouring a third cup of milk into a monkey's lungs and 2 weeks later saying, "Oh my God, we found milk in there." Of course you did. You put it there, and you are specifically only looking for short genetic milk sequences with your PCR test. Yet these foreign nucleic acid sequences, unsurprisingly, disappeared from the monkeys' bodies over the next few days through natural clearance mechanisms, despite not being given any vaccines or antiviral medication. So why are you taking them? SARS-CoV-2 RNA was only detected in a rectal swab from 1 out of the 8 animals on day 14, and no viral RNA was detected in whole blood at any time in any monkey throughout the study. No viral RNA was detected in the blood, aside from our moving nervous system fluids, which should be able to spread alleged respiratory virus particles from our lung tissue up to our brain and out to all the other connected organ of the body they allegedly never also spread to. And seeing as our digestive system doesn't connect our lungs to our rectum because it bypasses the lungs and starts at the stomach, the blood circulatory system is the only other way alleged respiratory virus particles could possibly get from our lungs to our rectum without infecting every other organ tissue in between on the way. It's the only other moving body transport system that connect the two. Again, this indicates that they were only finding the same introduced genetic material in the same places it had been introduced. The single positive rectal swab may have been a false positive, or the monkey had swallowed some of the introduced biological material. In not one case could they demonstrate that the postulated virus had any invasive characteristics. 4 of the monkeys were murdered and autopsied 4 days after inoculation with the foreign biological soup. 2 out of the 4 were reported as having small foci of consolidation in their lungs, or what you might call pneumonia from damaging their lungs. And the authors stated that the main histological lesion in the consolidated pulmonary tissues of both the young and aged animals involved the alveoli and bronchioles and consisted of areas with acute or more advanced DAD, diffuse alveolar damage. The histological features, meaning taking a wild guess what you're looking at under a microscope, were asserted to be characteristic of SARS-CoV-2, even though the same study says the other organs in these two macaques, as well as the respiratory tracts and the other organs of the other two animals, were normal. And that is because the top 3 slides across in their paper, A, B, and C, are consistent with pneumonitis, caused by pouring a liquid containing foreign biological material directly into the monkey's trachea while it was anesthetized. The histological appearance changes in the bottom 3 slides across, D, E, and F, simply depict inflammatory tissue cells, such as macrophages and neutrophils, as would be expected in such inflicted pneumonitis. No control experiments were performed, pouring a third cup of any other fluid down their trachea to see if the exact same pneumonia-causing cell tissue damage occurred. The alveoli are the end point of the respiratory system on the lungs and allegedly exchange oxygen and carbon dioxide in the bloodstream, sort of like the little green balls on the end of broccoli. And the bigger cells on the outer surface of those little balls that do this gas exchange are called type 1 pneumocytes. The paper claims SARS-CoV-2 antigen expression was detected in moderate numbers of type 1 pneumocytes and a few type 2 pneumocytes within foci of diffuse alveolar damage. The problem is, this was claimed through an immunohistochemistry staining technique that was based on a rabbit polyclonal antibody against SARS-CoV nucleoprotein. Why is that a problem? Because the supplier of this product, Sinobiological, admits IHC, FCM, IF, IP et al. Applications haven't been validated. Antibodies applications haven't been validated with corresponding virus-positive samples. And this tonic will make your hair grow back, even though it's never once been proven to do so and the guy selling it to you is still bald. In any case, this example can be used to expose the wider fallacy regarding antibodies as any evidence of viruses. Sinobiological states that the antibodies were the result of injecting their SARS-CoV nucleocapsid protein, his tag product, into rabbits. This nucleocapsid protein was in turn produced from a DNA sequence encoding the SARS-CoV isolate TOR2 nucleoprotein by use of a product on the market with testing in progress. We'll get into the so-called TOR2 protein in Part 2, but in summary, outside of a computer-simulated hypothetical in silico model, no protein has ever been shown to come from a virus, including the nucleocapsid protein in this case. It was baselessly asserted that they injected viral proteins into animals instead of just other regular foreign proteins, and in response, the animals produced other proteins that are claimed to be antibodies. A virus was neither shown to exist nor required to exist for this sort of pointless exercise. Again, we are told a specific viral protein like SARS-CoV-2 will make the body create a specific antibody protein that will only bind to that alleged SARS-2 virus protein and nothing else. Yet, the COVID-19 vaccine generated HIV antibody proteins in 100% of the volunteers injected with the University of Queensland candidate vaccine. Looks like there's a major problem with the HIV hypothesis. If you can have so-called disease-defining HIV antibodies without any possible HIV virus to trigger the body to create them. Getting back to the study at hand, it did not follow the scientific method as it lacked controls. Not only were there no control experiments, but they didn't even demonstrate animal-to-animal disease transmission from the waterboarded monkeys allegedly infected to healthy monkeys. If these viruses are so airborne and infectious, why do they have to inject monkeys or pour fluids down their nose and throats? Why not simply aerosolize a sample into the animal cages so they inhale it? Once again, such experiments are avoided in order for the virologists not to refute themselves with regard to claims of contagion involving the imagined particles. Which brings us to the virus quantity paradox. We are led to believe that inside a host such as a human, the viral particles are produced in such great numbers that they can rupture the very cells containing them, while at the same time, paradoxically, they are present in such tiny amounts that virologists say they can't be seen in any patient specimens. Apparently, with regard to the alleged SARS-CoV-2 particle, it has somehow been magically calculated that one sneeze of a COVID-19 patient contains 200 million viruses. However, if we obtain a physically larger sample directly from a subject's nose or lungs, equivalent to hundreds of sneezes, there are precisely 0 to be found, rather than billions, as claimed. So virologists pull the magic rabbit out of their hat by claiming they are invisible while grown in your lung sputum and can only become visible once grown in their magic tissue cultures.
Now, here's the first question. Since the viral theory is I breathe in this virus, it uses my lungs as a culture, right? And so then it grows, and then it's teeming with viruses that get spewed out in the world. Why can't you find it in your lungs? It's a culture.
Oh, your point is if you do a direct electron microscopy of the fluid, you won't see this.
Never.
Okay, yeah, I see.
Never. Not once. We asked a guy who was 20 years, senior virologist at Wuhan Institute of Virology, 20 years head of Yale Virology Laboratory. Can you find SARS-CoV-2 in any fluid of any person you say has COVID? He said no. Why not? Because there's not enough virus to find. Okay, we asked him, what about if you put 10 people with supposed COVID and you mix their bronchial fluid together? Is there enough? No. 100 people? No. 1,000 people? No, not enough to see. 10,000 people, you mix their fluid together. Would you then have enough virus to see? And he said no, and then he wouldn't answer anymore. So,
but he claims what we see on the EM, on the electron microscopy slides, are in fact virus particles. The little bleb
what
things.
He says is you have to culture them. There is no virus to see in your lungs,
right,
or in your snot.
Enough that they'll admit
anywhere.
Yeah.
Both Dr. Mark Bailey and Dr. John Bevan Smith also destroyed this virological nonsense in their paper, The COVID-19 Fraud and War on Humanity. So we are supposed to believe that the human respiratory tract, which is lined with the alleged perfect host cells, does not produce enough viruses for them to be seen. But a test tube experiment involving tissues from a different species and cell type does? Pfizer suggests to the layperson that the immune system reacts to the injury of these bodily cells by revving up, but did not cite any scientific evidence for this imaginative claim. In fact, aside from poisoning, nobody can prove there's anything else to even be immune from. The 4th edition of Medical Microbiology has offered up 4 hypothetical pathogenic mechanisms. Hypothetical means an assumption but not fact that can be tested by further investigation. And pathogenic means mechanisms that cause a diseased or morbid condition that may result in cell damage and death, or they may not. But again, even if these speculative measures were at play, it would require enormous numbers of cells to be affected to produce symptoms, just like it would take enormous amounts of institutions worldwide to keep this fraud going. But unlike the very real institutional viruses of this world, We can never find these virus particles in our own cells or tissues because virology has a very real habit and unique way of diverting attention away from such aspects that raise doubts about its phantasmal model of disease. End of part 1 and the end of virology. Part 2. The fraud used to propagate the COVID-19 pandemic.
It's done in a single individual, a single human being. All right. There were 44 cases of pneumonia in December, I guess it was, of 2019. And the World Health Organization office there in China cataloged these 44 people of pneumonia that they had no explanation for. And this is in a country that has about 1 million pneumonias every year. All right. What is so important about 44 cases? They say don't know what's causing it. Lots of things can cause pneumonia. Chemicals can. Malnutrition can. Drugs can. Being old can. Being in a hospital can. Chronic diseases. I mean, there's all kinds of things that can cause pneumonia. And with this one guy, okay, so now they have this one guy in a country of over 1 billion people. So then they did this massive, what I call a dragnet, for all of the RNA. They bound and determined to find a virus as a cause for this guy. So they did this dragnet for all of this RNA, millions of little strands of RNA in this person, using technology that's called metatranscriptomics. It's one of these gene things. It's technology-driven stuff. All this technology is technology-driven, not science-driven. So I got this person's lung sample of his lung. They do all this stuff. They have all this fancy equipment. And they zero down to get all these segments. Then they have a computer that stitches all these little segments, these little fragments of RNA together out of millions and millions and billions of these things. All right? And they came up with a sequence. And then they decided that they had discovered this virus, even though they never touched the virus at all. And they said that was the cause of this guy's pneumonia.
In the COVID-19 fraud and war on humanity, Dr. Mark Bailey and Dr. John Bevan Smith documented the invention of SARS-CoV-2 by Fan Wu's team, who assembled an in silico genome from genetic fragments of unknown provenance found in the crude lung washings of a single case and documented in a new coronavirus associated with human respiratory disease in China. In silico simply means in or on a computer, done or produced by using computer software or simulation, not what's actually in that patient's lung fluid in reality. It just means taking the tens of millions of genetic sequences in that one patient's sputum sample and having the computer rearrange whatever small fragments it needs from the entire bulk sputum sample, as we covered in Part 1 on metagenomics, into a much, much smaller 30,000 genome sequence. They want to accredit to either matching an old in silico computer-created virus genome like SARS-CoV-1, or even to creating a new computer in silico novel virus template like SARS-CoV-2 by using software such as the Trimomatic program, Megahit, Trinity, Blast-N, Diamond Blast X, RSEM, Bowtie 2, or in Fan Wu's case, all of the above together. Italian plumber, in silico Italian plumber. The claim that anyone can declare they identified a new RNA virus strain from the family Coronaviridae, which is designated here WH-human1 coronavirus, from a single human subject diagnosed with pneumonia is farcical in itself. The authors tried to justify this by stating, although the isolation of the virus from only a single patient is not sufficient to conclude that it caused these respiratory symptoms, our findings have been independently corroborated in further patients in a separate study. Firstly, there was no isolation of any alleged virus due to using metagenomic RNA sequencing, as taking an entire bulk sputum sample for metagenomic sequencing is not isolation of only one minute portion of unique identical particles out of that entire bulk sample, alone and apart from the rest of the sample, as covered ad nauseum in Part 1. Secondly, their claim of being independently corroborated is a reference to the February 2020 paper of Peng Zhao et al., a pneumonia outbreak associated with a new coronavirus of probable but not proven bat origin, which cannot corroborate anything, and we'll dissect the fallacy of that paper later in this film. All that can be said about Fan Wu's paper is that if circular reasoning is employed, then finding similar genetic sequences on more than one occasion is seen as confirmation of a virus.
See, it's not different. It's the same, just changed.
Look, I work for the phone company. I've had a lot of experience with semantics, so don't try to lure me into some maze of circular logic.
The GEISAID database is the treasure chest of this virological nonsense, and by August 29, 2022, had over 12.8 million claims of having found SARS-CoV-2, and as of August 30th, 2023, nearly 16 million claims of HCoV-19 variants. Better go get your 16 millionth booster just to be safe. However, none of them can point to an actual virus. They are simply calling bingo by assembling similar sequences which they have aligned with Fan Wu et al. And other previous assemblies. No actual virus required. Febrile is a fancy word for characterized by or accompanied by a fever. It should be noted that while the Fan Wu paper does not make any pronouncement as to the cause of any case of pneumonia or acute febrile respiratory syndromes, stating, "Here we report on a series of cases caused by an unidentified pneumonia disease outbreak in Wuhan." Even the general medical community acknowledges that no quote-unquote pathogen is identified in around half of these cases. Like the Liu Gaoshan paper, viral and bacterial etiology of acute febrile or fever-accompanying respiratory syndrome among patients in Qinghai, China, where 445 nasal swab specimens were taken from patients with the exact same acute febrile respiratory syndrome symptoms as Fan Wu's single patient, used real-time PCR, and yet only 225 out of 445 returned positive specimens, despite all having the same alleged viral or bacterial causing symptoms. So knowing that this science is as inaccurate as a coin toss or a spin on a roulette wheel, what reason did Fan Wu et al. Have to suspect that their patient was harboring a brand new virus? Apparently, because epidemiological investigations by the Wuhan Center for Disease Control and Prevention revealed that the patient worked at a local indoor seafood market. It would seem a very weak reason, given the fact that these wet markets are extremely common in China, and that despite the bat origin theories, Fan Wu et al. Reported no bats were available for sale. So, if they admitted no bats were sold at the market, why did they say their study was independently corroborated by another study claiming a new coronavirus of probable bat origin? And why did the media push this ridiculous story down our throats? In any case, Fan Wu's team collected a crude specimen of bronchoalveolar lavage fluid from their single patient. They do this by sticking a bronchoscope that pumps saline solution into your lungs and then sucks the fluid back out, along with some of the patient's crude lung fluid. They reported that total RNA was extracted from 200 microliters of bronchoalveolar lung fluid, or 200 millionths of a liter. Their methods section detailed that this was achieved using the RN Easy Plus Universal Mini Kit QIAGEN, i.e. through spin column centrifugation, which basically entails contaminating and destroying your crude lung sample by adding detergent, buffers, proteinases, or other enzymes, and heating it to high temperatures to destroy the cell membrane through lysis to release the RNA, then contaminating it more with an acidic buffer solution along with ethanol or isopropanol and spinning it through a silica membrane to make the RNA stick to the silica, and then washing that with a new acid buffer, more ethanol, isopropanol, and finally alcohol, and then adding more salt and heat, ensuring the sample is nothing like it was when it left your lungs. They claim that ribosomal RNA depletion was performed during library construction. Later, we'll get into why this is a dubious claim, as there remained a high, nearly 99% match with Homo sapiens RNA. But it is possible Fan Wu's team went through the American public school system and don't know the difference between a human being and an alleged virus particle.
We have to find a way to live with the Homo sapiens in a way where we don't, they don't affect us, we won't affect them, kind of like in a separation type of way.
Have you ever seen a Homo sapien?
You don't want to know. Yeah, I saw one once at the zoo.
Describe it.
Furry.
Yep.
Big. Gorilla looking. Homo sapiens. Let them die. Save the humans.
I don't know what a Homo sapien is. If they're going extinct, that's very sad, but at the end of the day, I don't care.
And neither does Fan Wu's team. Fan Wu's team then took that brew full of random fragments of genetic material and began shotgun sequencing that brew, shredding it up more into even shorter sequence lengths, averaging only 150 nucleotides long.
Shotgun sequencing is the method that was used by the Private Genome Project. Shotgun sequencing requires multiple copies of the genome, which are effectively blown up into millions of small fragments. Each fragment is then sequenced. The small fragments are assembled using an immense amount of computer power to match overlapping sections. The drawback of this method comes when dealing with repeat sequences. Often there is no way of knowing how long the repeat sequence is, or in which of the many different possible positions the fragments overlap. Even the incredibly powerful software used to shotgun sequence the human genome couldn't cope with this. So Celera, the private company which relied on this approach, had to use the public data to fill in the gaps left by the repeats.
Meaning to put sequences in there that aren't really there. This is done because expedient technology like Sanger or PCR can only read short DNA strands of 100 to 1,000 base pairs long, not 30,000 long, like an alleged virus particle. So any sequence in the brew larger than 1,000 base pairs long needs to be chopped up into small pieces and then read and assembled by a computer by finding ends of fragments with the same small sequences at the ends of other fragments and putting them together like a puzzle, whether those fragments came from the same one original genetic source or not. Remember from part 1, there are dozens of different genetic sources not only in tissue cultures, but also in a sputum sample. Once you blend them all up into tiny fragments, the computer can reassemble any of them it likes together to create something that was never really there in the first place, like an alleged virus, no different than Frankenstein, as both are fiction. Fan Wu's team also converted the RNA to DNA using a reverse transcriptase enzyme using Illumina minisec technology. In total, they generated more than 56.5 million of these short sequence reads from that one lung sample and then fed those sequences into both Megahit and Trinity software for de novo algorithm-based assembly into longer segments, like trying to put a puzzle together without the picture on the box. Contigs are just hypothetical overlapping long sequences created in a computer by assembling gel readings of thousands of tiny sequences whose ends have identical overlapping nucleotide sequences with the ends of other tiny sequences. It doesn't matter if those tiny sequences all came from multiple different genetic sources in the lung fluid sample. Once all broken down into 56 million pieces and mixed together, they can be pieced together into tens and tens of thousands of new contig genomes that weren't even there in the original lung fluid sample. It is fitting that the word contig starts with con, because this procedure is one giant con job. Through Megahit software, Fan Wu's team generated just over 384,000 different contig sequences, and the longest one generated was 30,474 nucleotides long, roughly in the ballpark for the alleged 30,000 long genome sequence of alleged SARS-type coronavirus particles. They don't say how many tens or hundreds of thousands weren't even remotely close. That sequence was said to have a nucleotide identity match of 89.1% to BAT SL-CoV-ZC45, another fictional construct. BAT in silico BAT. Ironically, they already admitted in the same paper there were no bats sold or eaten at the wet market they suspected, but facts don't matter to virologists. Remarkably, 89.1% is pretty close to the alleged 87% genomic match humans are supposed to share with a mouse, or maybe even a flying one. Hell, 89.1% is even closer to the alleged 90% genetic similarity between a human and a cat. Would you find a stray cat in your yard and say, "Honey, I found a lost child in the woods"? Well, that's what Fan Wu's team did. Even more comical, the other software program, Trinity, generated over 1.3 million different contigs out of the 56.5 million small random sequences. But the longest genome it could fictionally assemble was only 11,760 nucleotides long, only 1/3 of any alleged virus particle genome. In other words, they would never have even found the genome in the first place if they had just used the Trinity software platform alone. In true pseudoscience fashion, they just went with the Megahit program that was at least closer to partially seeming to confirm their theory, rather than investigating why the other Trinity program they rely on debunked it completely. That's like blaming this guy for stealing your car. The word virus suddenly appeared out of nowhere when they stated the genome sequence of this virus, as well as its termini, meaning boundary or border, were determined and confirmed by reverse transcription PCR. This is just more sleight of hand. PCR has no capacity or technical ability to either confirm or detect a full previously unknown genome. That is why using a PCR test to detect if you have an alleged virus is as comical as looking at one tiny fragment of one single white hair under a microscope to determine if you have been trampled by a white horse, a zebra, a unicorn, a pack of sled dog huskies, or even your old grandma for that matter. If you take 56 million fragments of common genetic material found in everyone's lungs at one time or another from various normal common genetic sources found in that lung fluid, and then computer assemble them into a fictional, contig, 30,000 nucleotide long virus genome, and then use PCR to look only for a few short sequences out of that hypothetical 30,000 nucleotide model, of course you're going to find it in seemingly alarming numbers out of all of the billions of suckers who ignorantly take these tests and the ignorant medical practitioners who use them. A PCR test is just amplifying and trying to detect minute 100 or so odd-long base-paired nucleotide fragments of unknown origin in your fluids or sputum and match them to the exact same sized minute nucleotide fragments of unknown origin that they originally took from someone with lung fluids just like yours to computer contig assemble a much larger fictional genome in the first place. Sometimes they match, sometimes they don't. Which is why you can PCR test positive without actually being sick. Asymptomatic does not mean an infection that magically makes everybody else sick, but not you somehow. It simply means PCR found the tiny, normal, common genetic material in your lungs it was looking for, but it obviously has nothing to do with actual illness symptoms. The Nobel Prize-winning biochemist Dr. Kary Mullis, the inventor of PCR, said it himself.
And with PCR, if you do it well, you can find almost anything in anybody. It starts making you believe in the sort of Buddhist notion that everything is contained in everything else, right? I mean, because if you can amplify one single molecule up to something that you can really measure, which PCR can do, then there's just very few molecules that you don't have at least one single one of them in your body, okay? It doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything like that. It doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything like that.
Back to Fan Wu's team using reverse transcription PCR to determine and confirm the genome sequence of an alleged brand new novel coronavirus. As PCR expert, Professor Stephen Buston, editor of the PCR Revolution, has explained, PCR requires you to know what the sequence of your target is. So once you know that there's something in your sample, then you would try to isolate it, yes. And then once you've isolated it, then you sequence it again, or PCR it up. In other words, PCR itself cannot identify the origins of the sequences, and the methodology of Fan Wu's team did not establish the origin of their described sequences. Yet, in the very next sentence, they announced to the world that this virus strain was designated as WH-human1 coronavirus, WHCV, later 2019 nCoV, and its whole genome sequence of 29,903 nucleotides was put into the GenBank database by Fan Wu's team as a novel, brand new virus. What new virus? We need to pause at this point, as it is where the fraudulent virus, soon to be renamed SARS-CoV-2, was invented out of thin air, a virus that the World Health Organization claims, with no evidential support whatsoever, is the causative agent of COVID-19, itself just a farcical rebranding of the same exact generic flu detox symptoms that have been around since the dawn of mankind. For it is this genome that was submitted to GenBank on the 5th of January 2020 that was seized on by Christian Drosten et al., never forget that name and face, to help produce their phony PCR protocol assay sequences. In detection of 2019 novel coronavirus 2019 nCoV by real-time PCR, Drosten's team just took Fan Wu's 29,903 nucleotide-long sequence genome, assembled out of 56 million blended up normal genetic material found in patient Zero's lung fluid, selected a few small segments out of that 30,000 long Frankenstein sequence, and created the probes and primers for the PCR test, which in turn were published with indecent haste by the World Health Organization for all the world to use, thereby turning WH-Human1 into the world's reference genome for a claimed pathogen. It is this invention that is responsible for the whole bag of destructive tricks imposed upon the world following the announcement of the pandemic by the WHO on the 11th of March 2020.
WHO has been assessing this outbreak around the clock, and we're deeply concerned both by the alarming levels of spread and severity and by the alarming levels of inaction. We have therefore made the assessment that COVID-19 can be characterized as a pandemic. Pandemic is not a word to use lightly or carelessly. It's a word that, if misused, can cause unreasonable fear, leading to unnecessary suffering and death.
However, anyone paying attention can see that there is no evidence whatsoever of a virus in the Fan Wu paper. A virus is claimed to be a tiny, replication-competent, obligate intracellular parasite consisting of a genome surrounded by a proteinaceous coat. That's what the British Society for Immunology tries to assure us, and it is alleged to be an infectious particle that causes disease in a host. All Fan Wu's team really had was one single 41-year-old man with severe respiratory syndrome, fever, dizziness, and a cough, what we call pneumonia, and a software-assembled model genome made from sequences of unestablished origin found in a single sample of the man's lung washings. That's it. To make it appear legitimate, they stated the Viral Genome Organization of WHCV was determined by sequence alignment to two representative members of the genus Beta coronavirus, a coronavirus associated with humans, SARS-CoV-2, GenBank number AY274119, and a coronavirus associated with bats, BAT SL-CoV-ZC45, GenBank number MG772933. But when that was the only genome in the GenBank database that kind of matched their computer-built fictional virus genome, they employed the pseudoscience known as confirmation bias. Ignore what the facts say, and instead go with what confirms your virus origins belief. As Dr. Mark Bailey points out, both of these alleged SARS-CoV-2 and BAT SL-CoV-ZC45 genomes are also simply in silico hypothetical computer constructs that have never been proven to exist in their entirety in nature, let alone been shown to come from inside an alleged virus. They're both as fictional as Batman. For example, BAT SL-CoV-ZC45 was invented in 2018 in a paper entitled Genomic Characterization and Infectivity of a Novel SARS-like, but not SARS coronavirus in Chinese bats. By the process of 19 degenerated PCR primer pairs, designed by multiple alignments of available SARS-CoV and BAT SL-CoV sequences deposited in GenBank. The virus genomes have become what is possibly the greatest illusion in virology, an illusion which propagates a false belief that viruses are indeed being shown to exist. The virologists themselves don't seem to appreciate the fatal flaw in their own methodologies, even when they state it themselves in papers like A Complete Protocol for Whole Genome Sequencing of Virus from Clinical Samples from 2019. Three main methods based on HTS, or high throughput sequencing, are currently used for viral whole genome sequencing: metagenomic sequencing, target enrichment sequencing, and PCR amplicon sequencing, each showing benefits and drawbacks. In metagenomic sequencing, total DNA and/or RNA from a sample, including host but also bacteria, viruses, and fungi, is extracted and sequenced. It is a simple and cost-effective approach, and it is the only approach not requiring reference sequences. So here they're admitting they're using DNA or RNA from you, the host, plus bacteria and fungi in your lung sample to help make their virus genome. Again, from part 1, that is not isolation and purification of a virus. That is Dr. Frankenstein. Instead, the other two high-throughput sequencing approaches, target enrichment and amplicon sequencing, both depend on reference information to design baits or primers. The limitation of metagenomic sequencing is that it requires a very high sequencing depth to obtain enough viral genome material. But as Dr. Mark Bailey and his rapidly growing list of colleagues keep pointing out to the seemingly deaf ears in the medical establishment, the more important limitation with all three methods of so-called viral sequencing is that the process itself does not determine the provenance, meaning origins, of the genetic fragments out of all of the various garbage in a sick person's lungs. So how can any of them be used to establish the sequence of a previously unknown genome, like Fan Wu's alleged new coronavirus?
They'll take, fluids from a sick host, from a sick patient, and they sometimes centrifuge it a little bit to break up some of the bigger particles, but they never separate everything. So there's always going to be some sort of bacteria, other microorganisms, host material. There could even be potentially what they call microvesicular bodies like exosomes. They're guaranteed to be in that sample.
And then my understanding is they take the kind of runoff of that culture and they put it through a metagenomic transcription program like BLAST, and they assemble a theoretical, an in silico virus, a theoretical computer-generated virus. It's not a virus, it's a computer code. And then that's what they test for.
Yeah, exactly. They test for these fragments that are said to belong to this theoretical genome.
Which is said to represent an actual genome, but no one has that model. No one has an actual genome to measure against, to compare it to.
Exactly. Well, because every single one of these genomes, when you trace the references back, they all come from an unpurified source. It's always going to come from some sort of tissue or cell culture. They're not, ideally what you would do if you were going to try and find a virus in someone, right? You would take their fluids. Like if I was sick, you'd take my lung fluid, let's say, and you would try to separate out the particles within that fluid that you believe is a virus. So then you have just the virus particles, nothing else, no bacteria, no host materials, no exosomes, no microvesicular bodies, whatever, can't pronounce it. You would have just those particles, and with those, then you can, test. Well, you can biochemically characterize them. You can test. You can see if you can infect animals. They don't ever do that.
So they claim to have a photo of a virus, but they can't pick that thing up with tweezers and actually sequence that, slice it up and sequence it. So instead they do the exact opposite thing, is they take this mishmash of countless ingredients from an impure patient sample that's never ever been separated, and then they crush that all together, and then their computer program builds up a concept.
Bingo.
To be perfectly clear here, we are not talking about situations where the provenance or origins of the sequence can be independently verified. For example, physically isolated bacterial cells, which is an easily manageable task, even for undergraduate medical students. Additionally, it is nonsensical to arbitrarily declare that sequences are somehow viral by a process of elimination, that is, based on the fact that they do not have a previously conflicting assignation on the genetic databanks. Why not assume they're unicorn sequences? They're not in the databanks either. As Mike Stone just pointed out, none of the virologists are demonstrating that the sequences are viral in nature when they assemble the very first template and declare they have discovered a pathogenic virus. That includes the 2 fictional human and bat virus genomes Fan Wu's team found in the databases, made the exact same way as his fictional virus genome. And some future virology team will do the same thing with the lung fluid of someone with pneumonia, computer assemble the 56 million meaningless fragments into a 30,000 long genome, find an 88% match to Fan Wu's fictional virus in the database, and call it SARS-3. And on it'll go like a broken record. At no stage are any virologists isolating and purifying these alleged viral particles and chemically and genetically characterizing them to at least try and either prove or disprove their relationship with the computer-assembled genomic sequences they are allegedly made of. If I told you your body was made out of Legos and not biological tissue, wouldn't you demand that I prove that to you by comparing Legos to your tissue sample to see if they are made of the same thing? And yet, the first invented Lego-assembled de novo or new virus genome becomes the touchstone with which other virus hunters will align their own in silico computer genomes or design confirmatory PCR protocols, metaphorically looking for small chains of fictional Lego blocks, not real-life virus genome sequences. We would like to see proof that virologists even have any laboratory techniques that even can confirm the existence of a 30,000 long RNA strand in any of their samples. With DNA, pulse field gradient gel electrophoresis can easily confirm a nucleic acid sequence strand of that length, but it still can't tell you what that sequence is composed of, viral or other, just how long it is. It can, however, check that your sample really was that long and not made to that length inappropriately with Frankenstein in silico techniques. However, with RNA, this technique is not thought possible for such long strands. So how could it even accurately or reliably check if a 30,000 long piece of RNA was even present in the sample? So when they say they've found the 29,000 plus long RNA viral genome sequence for SARS-CoV-2 on their in silico computer simulation, they only have a hypothetical model and can't actually independently verify that the entire sequence even exists in nature. In any case, these computer genome simulations remain a distraction because even if they did exist in nature, which they don't, as we also covered ad nauseum in part 1, the virologists would still have to demonstrate that this genome sequence belongs to a disease-causing, replication-competent particle that can make a person ill and not just claim that it does through fictional Hollywood movies. And yet, even though not thought technically possible, Dr. Mark Bailey had a long email exchange with evolutionary biologist Zachary Ardern from the Wellcome Sanger Institute, who suggested that long-read RNA sequences, as opposed to only shotgun sequencing, provided the necessary proof of the existence of SARS-CoV-2. Ardern referred to this April 2022 publication in Frontiers. Long-read RNA sequencing identifies polyadenylation elongation and differential transcript usage of host transcripts during SARS-CoV-2 in vitro, or in a test tube, not in vivo, in a living organism, infection, involving implementing RNA sequencing using Oxford Nanopore Technologies ONT long reads, claiming that it confirmed the validity of the virus genomes that had been previously artificially constructed through short-read shotgun sequencing. The preferred study describes an experiment comparing responses between various SARS-CoV-2 infected and mock-infected cell lines. But infected by what? The experimental cells were alleged to be infected with SARS-CoV-2 Australia virus, Australia VIC01 2020, claimed by its authors, Leon Kelly et al., to be a virus isolate, when isolation of a virus was never demonstrated in Kelly's original paper of that alleged Australia virus. Kelly's team claimed they isolated SARS-CoV-2 from a 58-year-old man from Wuhan, China, who arrived in Melbourne on the 19th of January 2020 with a fever, cough, and difficulty breathing. So they had to be looking for an alleged coronavirus, named after its unique appearance from other alleged virus particles because of the spikes sticking off its surface, like the sun's corona. But they admit they didn't find anything like that in their cultures. Instead, they found spherical-looking particles of at least nearly the same size as an alleged coronavirus, about 90 to 110 nanometers in diameter, but then admitted, following several failures to recover virions with the characteristic fringe of surface spike proteins, it was found that adding trypsin to the cell culture medium immediately improved virion morphology, meaning appearance. Meaning the particles didn't even look anything like alleged coronavirus particles until after the added enzyme trypsin digested the outer proteins to make them look more like an alleged coronavirus. As usual, there was no valid control experiment performed with another human specimen to see if the trypsin and culture procedure gave the same resulting particles. You may ask, well, if there were no coronavirus particles in this 58-year-old man's sample, how did Cayley's team sequence his genetic material and get a greater than 99.99% sequence identity match with other publicly available genomes in the database, like Fan Wu's, from totally different people? Isn't that a slam dunk proof they must have the same virus? No. Because when we look at the details in the Medical Journal of Australia of what Cayley's team actually did, we see that they prepared an Illumina library with a Nextera XT kit to break all of his genetic material down into tiny 75 base pair end fragments, and were then able to get 30 million fragment reads using CISPA, sequence-independent single primer amplification, which, unlike PCR, requires target sequence modification to tell it what tiny sequence to build out of that entire genetic muck. You have to tell it what sequences you want it to make, and it does it. Then Cayley's team took those 30 million purposely CISPA tailor-made tiny sequences, chopped into millions of tiny Lego pieces out of all of the larger crap in this guy's lungs that should fit perfectly like Legos into the larger genome they were trying to build, and used both de novo assembly, not telling the computer what long contig sequences to assemble them into, but seeing what it comes up with randomly on its own, and more importantly, also alignment to reference sequencing, telling the predictive algorithm software to make an assembly that matches already existing genomes like Fan Wu's from the database. Then they mixed the best results from both of those techniques together to get a 99.99% match to what they wanted to find. That is why even though Fan Wu's team had 56.6 million sequences to play with rather than Cayley's 30 million, Fan Wu only used random de novo sequencing with no assigned target genomes, and then afterwards checked if any of those 1.6 million total contig assemblies matched any already existing genomes, and only got one with an 88% match to two already existing database genomes. It makes you wonder if Fan Wu had used 75 nucleotide long segments and the Cayley dual method approach, if they could have got a 99% match to some other alleged genomes. It's like telling the computer to assemble these random Lego blocks into this castle, which is what Cayley's team did, or telling the computer to take these blocks and assemble hundreds of thousands of random castles out of them, and then check the castle database to see if any of them were at least kind of close in structure to this castle, which is what Fan Wu's team did. And because 99.99% is not 100%,
missed it by that much.
You now have Cayley's Beta Cov Australia VIC01 2020 genome to add to the other 16 million variants of HCoV-19, aka SARS-CoV-2, created the exact same bogus way. They are all doing the same scientifically flawed process on an infinite loop of circular reasoning. There is no independent validation of their result, and they have no valid controls in place. All they can say is that if you go on a fishing trip for genetic sequences in mixed biological soups, then you can produce such results. Hence, the evolutionary biologist Zachary Ardern's argument relied on the fraudulent product of a fraudulent long RNA sequence read experiment, as just described, being compared with a mock infection, where the former is invalidated by the misleading declaration of virus isolation, and the latter mock infection invalidates itself, as covered in Part 1, as the virologists have changed its definition to allow too many other variables to be altered to be a scientifically valid control. Since phosphate buffered saline solution contains 0 of the dozens of various rotting material sources and toxic chemical additives found in a sputum soup sample prepared for any such dodgy, invalid control experiment. Obtaining meaningless longer reads does not change these foundational issues. Ardern was asserting that variations in monitored sequences and proteins over time represented evidence of an evolving virus. Just like this butterfly must be evidence of a caterpillar virus, because there are apparently no other possible reasons protein sequences might evolve into other protein sequences over time, or extreme changes in environmental conditions. Let alone virologists chemically chopping up one thing into millions of pieces and purposely rearranging it all into something that wasn't even there in the first place. As virologist Dr. Stefan Lanka explains, these particles here are shown to us as viruses, but what are they really? They take broken down tissue, isolate this tissue from everything around the core that can still survive, and chunks of the tissue develop little fingers, spikes or crowns very quickly, like an amoeba that wants to rebuild the tissue again. That's why everyone who works with these cell cultures and test tubes has to constantly separate them again and again. These little chunks of tissue are misinterpreted as cells, And these little fingers with which it drags itself along to try and rebuild the tissue, these are called the villi, of which a cross-section in the electron microscope underneath is presented to us as viruses. It's that simple. It's hocus pocus zimzalabim. We see that the virologists, later we will learn why, are completely anti-scientific because they never on any level of their work do or document control experiments. For example, they could have cut through chunks at the edge or in the middle. In the middle, the diameter appears large. At the edge, it is smaller, like this. And if I am outside the chunks of tissue, then nothing would appear. And those on the edge are the only photos they are constantly presenting to us. You don't even find areas where you just have some spherical particles. If you look, the rest of the tissue is still there. You have an anti-scientific error. This pseudoscience is the basis of their actions. I can only call something scientific if it hasn't been refuted or falsified. And you didn't do that at any level of your research. In the end, Zachary Ardern is just another victim of virology's deception through their specious attachment of the word viral to both these visual particles of unknown origin and these computer-assembled protein sequences. Despite the fact that when all such tens of millions of sequences and proteins were originally detected in sputum samples and tissue culture experiments, they were not shown to belong to pathogenic viruses, the claim by virologists that they are viral still continues to this day. Virologist Dr. Stefan Lanka summed up virology perfectly in 5 seconds.
What they have in reality in their test tubes, right? They have nothing.
Along the same lines, and a few months after that awkward exchange with Ardern, who couldn't give any real scientific evidence, so he simply ran out of unpaid personal time to discuss the most important issue of our time, the pathologist/virologist Dr. Sin-Hang Lee published his preprint paper, Implementation of the ECDC WHO Recommendation for Molecular Diagnosis of SARS-CoV-2 Omicron Subvariants and Its Challenges. I'll say, it is challenging to detect a subvariant mutation of something that never existed in the first place, but that never stops virologists. Lee claimed that his paper provided irrefutable Sanger sequencing evidence that the virus SARS-CoV-2 exists and keeps mutating, with an open invitation to critique and challenge his data and interpretation online in the open, where other scientists can join in for the debate. So Dr. Mark Bailey responded, "To expose the problems of virology, it is crucial to examine the methodology section of any publication, and in Sin Lee's case, it is no different." In his material and methods section, Lee stated that 5 selected nasopharyngeal swab specimens collected from non-hospitalized patients with respiratory infection were confirmed to be true positive for SARS-CoV-2 Omicron variant by Sanger sequencing. The first problem is, aside from wondering why not one of these 5 people were hospitalized if they had a deadly virus variant, here again we are straight into the deep end of virology's circular reasoning. The virus has been confirmed to exist on the basis of detected sequences from some nasopharyngeal swabs. There is nowhere in his paper that any evidence is provided for the existence of an actual virus, that is, a tiny particle that acts as an obligate intracellular replication-competent parasite that is capable of causing disease in a host. The second problem is that a common challenge of DNA or RNA sequencing with the Sanger method is the deteriorating quality of sequencing traces after 700 and 900 bases. So current methods can directly sequence only relatively short 300 to 1,000 nucleotide long fragments in a single reaction. Certainly not 30,000 nucleotide long alleged virus genomes or alleged mutated virus variant genomes. So the obstacle to sequencing fragments above this size limit is the insufficient power of separation for resolving large fragments that differ in length by even only one nucleotide. So in Lee's irrefutable Sanger sequencing evidence wisdom, he chose anyway to just pick 3 small genomic targets for these 5 nasal swabs, and found matches to 3 different, smaller target variant sequence matches, 3 of them matching Omicron BA2, 1 matching Omicron BA4 or BA5, close enough either way, despite just 1 nucleotide difference being a problem with this method, and 1 matching Omicron BA1. Out of the nearly 16 million target choices to choose from in the computer database. What a miracle! Lee's claim that the 5 selective nasopharyngeal specimens were confirmed to be true positives for SARS-CoV-2 Omicron variant simply means some small sequences in these 5 people's nasal booger samples match some other small sequences that were previously computer concocted out of someone else's lung snot sample and deposited on genetic databases and fraudulently declared to be viral, whether actually viral or not, and were simply being detected again by another method other than PCR or another. Is that why none of them were sick in hospital, despite having 4 deadly variants of this virus? It doesn't make any difference which sequencing technique is used to detect a genomic sequence, because the crucial issue is none of them can determine either the origin or the clinical relevance of any of the sequences they can detect. They literally have no idea if those detected sequences came from a virus or just from normal genetic material in your sputum sample, and they have no idea or proof that if that genetic sequence was found in your sputum sample, it would somehow cause you physical illness or not. So Dr. Mark Bailey rightly pointed out to Sin-Heng Lee, those of us that dispute the virus narrative point out that no RNA or DNA sequences have ever been shown to come from inside any specific identifiable particle that fulfills the definition of a virus. A tiny particle that acts as an obligate intracellular replication competent parasite that is capable of causing disease in a host. Thus, all RNAs can only be said to be expressed by a known organism, introduced artificially, e.g. synthetic mRNA injections, or be of unknown provenance or origin. The mutations or variants only exist within in silico computer models that have not been shown to be independent entities in nature. There are other reasons why RNA sequences can and do vary in dynamic biological systems, and Dr. Bailey can't imagine that any virologist would disagree with this fact. Simply detecting RNAs is not enough to draw conclusions about their provenance or origin. Other experiments are required to make this determination. Not just finding 3 small specific RNA genomic targets through Sanger sequencing with no proof of where they come from or if they are disease-causing. No amount of genomic or proteomic technology can escape the fact that with regard to such data being supposed evidence of viruses, It's simply a bad case of turtles all the way down. It's hard to imagine how an intelligent, seasoned pathologist/virologist like Sin-Hang Lee can't see this simple fallacy. But as Upton Sinclair said, it is difficult to get a man to understand something when his salary depends on his not understanding it. Scientists tend to agree 100% with the people who are funding them. You might be saying, we don't want to jump the gun here. Just because we caught Fan Wu's team building a fake in silico virus genome, how do we know the other viruses in the database that his fake genome closely matched aren't real themselves, like bat SL-CoV-ZC45? Isn't that a real thing? Over 60 viruses have been identified in bats, right? That's what the internet says. Bat SL-CoV-ZC45 comes from a September 2018 paper by Dan Hu et al. Called Genomic Characterization and Infectivity of a Novel SARS-like, but not SARS, coronavirus in Chinese bats. It was purported to come from the intestinal tissue of a bat that was captured in Zhejiang Province, China. Not to beat a dead bat yet again, but again from part 1, SARS is alleged to be a respiratory lung virus connected to the respiratory system and the lungs, which are not connected to the digestive system and intestines. So finding an alleged SARS-like lung virus in a bat's intestines should be your first clue that something is wrong. Clue number 2. All bats appeared healthy and had no obvious clinical signs of viral illness at capture. Wouldn't you want bats that seem sick with a virus? Clue number 3. Despite all 334 bats being completely healthy at capture, after being given a bogus screening for cov-RNA using a pan-coronavirus reverse transcription PCR test, as covered in part 1, it turned out that only 26.6% of them, or 89 out of 334 bats, actually were extremely sick with a deadly bat coronavirus after all, despite all 334 being completely healthy. Who knew? Clue number 4. Despite the failed isolation of the quote infectious virus, meaning they weren't infectious or harmful particles after all, or they wouldn't have failed to destroy the culture tissue, from PCR-positive samples of ground-up bat intestines put into Vero E6 monkey kidney tissue cells, remember from part 1, the only reason they even use Vero E6 green monkey kidney tissue in cell cultures in the first place is because almost anything can easily break it apart and cause cytopathic effect due to its aneuploid abnormal number of unstable chromosomes. But not this alleged dangerous infectious bat virus, though, whose alleged 89% identical cousin SARS-CoV-2 we locked the whole world down over, even though it could only destroy Vero E6 monkey tissue and not 5 other tissue types, whereas this one couldn't even do that. So obviously they must be nearly identical to each other, right? But since the alleged bat virus failed to break down unstable green monkey kidney tissue in a cell culture, like alleged virus particles are supposed to and alleged to do, they instead attempted to isolate the non-existent bat virus from suckling newborn baby rats by infecting them with the ground-up bat intestine tissue samples that PCR tested positive for the coronavirus. Scientifically, 3-day-old suckling rats were intracerebrally inoculated with 20 microliters of volume-grinding supernatant of ZC45 intestinal tissue. In plain English, they killed and cut open some bats, grounded up 20 microliters of dead bat intestinal tissue and injected that directly into the brains of only 3-day-old baby suckling rats. By weight, it would be the equivalent of injecting several hundred milliliters of ground-up intestines from fresh human corpses directly into a human brain. The nonsense of injecting such biological tissue directly into the brains of inbred, compromised neonatal animals shouldn't need any further explanation. As is typical in virology experiments, there was no control group where similar biological material said not to contain the virus was injected directly into the brains of other baby rats.
Okay, last time. This is drugs. This is your brain on drugs. Any questions?
Actually, yes. Is it just possible that taking ground-up bat intestines and adding the highly toxic poisonous drugs amikacin, vancomycin, and nystatin, injecting those drugs into the brains of newborn suckling rats, then killing them, grinding up their brains, and adding more toxic formalin or formaldehyde, alcohol, paraffin, aka kerosene wax from petroleum, toxic color dye irritant, hematoxylin, and eosin, a fluorescent acid used in paints, then adding more toxic dialdehyde, highly poisonous osmium tetroxide dye, ethanol, epoxy resin, chemically toxic and radioactive uranyl acetate from uranium, and finally, poisonous lead citrate might be the actual reason their brain tissue ends up looking like this under an electron microscope and not because of an alleged virus particle. They reported that suspected viral particles were seen in some of the rat brains, but at no point did they demonstrate the composition or biological function of such observed suspected viral particles in any of their slides. Electron microscopy is no different than a psychiatrist's Rorschach blot test. Just because you choose to see a bat here isn't proof there's really a bat here. Additionally, infection was declared on the basis of positive RT-PCR tests that detected the same RNA sequences in the baby rats at the time of their sacrifice as had been injected into them recently. Obviously not something that requires the existence of a virus. Imagine mushing up 3 Twinkies, injecting them directly into a newborn baby's brain, killing that baby, and then testing its brain tissue for glucose molecules. Would you be shocked to find them there? How would you know if that glucose was from the baby's own brain glucose or the glucose in the 3 Twinkies you just injected? At least if you tested for yellow dye number 5 and found it, you'd know it was from the Twinkies. But would you say that is proof that baby caught a Twinkie virus? Of course not. So without physically isolating any alleged virus particles, they proceeded to homogenize, centrifuge, and filter the bat intestinal samples before declaring the viral RNA was extracted with the viral RNA minikit QIAGEN, according to the manufacturer's recommendations. A reverse transcription step then took place before PCR amplification of their brew. They claimed to sequence the full genome of SL-CoV-ZC45 through 19 degenerated PCR primer pairs, designed by multiple alignment of available SARS-CoV and BAT SL-CoV sequences deposited in GenBank, targeting almost the full-length genome. We couldn't find a Ferrari, so we built almost a full one out of old spare parts. Well, kind of. In other words, just like all of the others, their declaration of discovering a viral genome was based not on direct evidence of a virus, but on detection of sequences of unestablished provenance aligned to yet more hypothetical virus templates. It was not disclosed how much PCR amplification took place at this step, but the RT-PCR screening step involved a first round of 40 cycles, followed by a second round of 30 cycles.
But also the PCR process makes de novo sequences. They make sequences which weren't there. The reason you know that is because if you do 10 cycles, you can only get 10% of the genome. If you do 40 cycles, you get 98% of the genome, which means that approximately 80% were made by the process, which is I mean, this is crazy. And yet we all believe it.
So it should be no surprise that a combination of 40 cycles and then another 30 cycles returned an 81% match to another fictional virus genome in the GenBank database. Of note, the bat virus story has been in place since the 2003 SARS outbreak, and apparently after thousands of years, the human race is now under constant threat from viruses percolating in Chinese bat caves. In 2005, the EcoHealth Alliance president, Dr. Peter Daszak, co-authored a paper that appeared in Science titled "Bats are natural reservoirs of SARS-like coronaviruses." In this study, Daszak and co. couldn't find any coronaviruses in their selection of bats through the usual fraudulent technique of observing in vitro cytopathic effects, stating that no virus has been isolated from fecal swabs of PCR-positive samples using Viro E6 cells. Again, even though PCR falsely convinced them that tiny genetic fragments in those bat poop samples must belong to viruses and not just normal bat poop, the alleged virus particles in that bat poop couldn't even break down the chromosome unstable aneuploid Viro E6 green monkey kidney tissue, like alleged virus particles are supposed to do in order to make you sick or even bother worrying about them. So instead, the complete genome sequence was determined directly from PCR products from just one of the fecal samples that contained relatively high levels of genetic material, or literally, a bunch of other shit to assemble a fictional virus genome out of, which is batshit crazy, pun intended. Of course, they were happily able to declare they had evidence of such viruses because once again, they used nonsensically high PCR cycle products, sometimes using 35 cycles and even a ludicrous 45 cycles for one of them. These were claimed to be viral sequences because within virology's circular reasoning, they found the very viral sequences that their PCR protocol was designed to detect. They duly warn the world that genetic diversity exists amongst zoonotic viruses in bats, increasing the possibility of variants crossing the species barrier and causing outbreaks of disease in human populations. Unfortunately, this zoonotic folklore has spread from the virology literature into the imagination of the public. Daszak is a keen promoter and financial benefactor of the bat virus story, and in 2015 advised his colleagues that in order to keep the revenue coming in, they would need to increase public understanding of the need for MCMs, or medical countermeasures, such as a pan-influenza or pan-coronavirus vaccine, or spying on you to make sure you take them. What a great guy. In any case, a branch of one of the imaginary coronavirus template trails leads back to one of the original claims made regarding the SARS-CoV genome, alleged to be the cause of the first SARS outbreak. In April 2003, Yijun Ruan et al. Submitted to GenBank their SARS coronavirus SIN 2500 complete genome, which became a session number AY283794.1. However, as usual, if you bother to actually read Ruan's paper in The Lancet, you find that this genome was invented, not by directly sequencing alleged virus particles, of course, but by sequencing the RNA in a viro cell culture experiment through both shotgun and specific priming approaches, with alignment to the mouse hepatitis virus genome sequence NC001846 as a backbone. Because that NC001846.1 genome was invented in turn in 1997 and was claimed to be derived from a virus that was obtained originally from Dr. Lawrence Sterman. The assertion that they even started their procedures with a virus in the first place appears to be based solely on Dr. Sterman's assurance that the sample he provided contained any such thing. It should be clear at this point that each alleged coronavirus genome has simply been templated against other previous so-called genomes in the database, without any of the virologists demonstrating that any of these sequences come from a virus particle and not just normal material found in biological samples. You could argue, as they might, that surely the very first coronavirus particle in the database was properly purified, isolated, and then chemically and genetically characterized. And that's the reason they can now assumptively take all of these time and money-saving sequencing and assembly shortcuts. It is thus instructive to go back to the purported first ever complete coronavirus genome to be published, which was the avian infectious bronchitis virus, IBV, by Bursnell et al. In 1987, and subsequently used by others as one of the original templates. As usual, they too did not sequence any postulated viral particles directly, but used 17 cDNA clones covering the 3' most 27,569 bases of the genome, noting that the clones have been derived from RNA isolated from gradient purified virus of the Baudet strain, Baudet and Hudson, 1937, then citing Brown and Bursnell 3 years earlier in 1984. Oh dear, here we go again. The cited Brown and Bursnell paper, Sequencing of Coronavirus IBV Genomic RNA, a 195 base open reading frame encoded by mRNA-B, states, the preparation of cDNA clones has been previously described in Brown and Bursnell 1984, then cites another one of their own publications from that year entitled Avian Infectious Bronchitis Virus Genomic RNA Contains Sequence Homologies at the Intergenic Boundaries. I know, George, but probably just a coincidence. In this paper, they claim that the IBF, or infectious bronchitis virus strain, Baudet, was grown in 11-day-old embryonated eggs. Virions, or virus particles, were isolated from allantoic fluid, meaning the outer fluid in part of an egg yolk, and purified by isopycnic centrifugation on sucrose gradients. Isopycnic just means marked by equal or constant density, or produced by a technique such as centrifugation, in which components of a mixture are separated on the basis of difference in density. But as was pointed out in Part 1, this technique is proclaimed to be meaningless by modern virologists because they now claim it is an almost impossible mission to separate extracellular vesicle particles from alleged virus particles using differential ultracentrifugation because they have the same size, dimension, and density, meaning they can't tell the virus particle from the milk particles in the same density layer, And nobody in their right mind would think that this is proof that a cherry tomato is made out of dish soap because they have the same density. Yet, at the exact same time, they cite this paper and methodology as proof of finding infectious bronchitis virus particles in egg yolk fluid, not even connected to the chicken fetus's lungs, but to collect liquid waste from its blood and digestive system. They can't have their cake and eat it too. No evidence was provided in any of these papers that they A. had purified anything, let alone virions, in the form of confirmatory electron micrographs, or B. performed any valid control experiments. All we can see is that because they are taught to either only cite previous papers and endless footnotes, and blindly carry out and repeat taught procedures, and not to actually bother to read these cited papers' methodology sections, or don't really scientifically question those procedures if they do read them, they just assume viruses were present in their culture mixture, and after centrifugation, they claim the detected RNA sequences were from these imagined viruses. And fittingly, when you click on the Hudson Baudet paper that these papers all lead back to as their origin, nailed it. Only kidding. That was their bogus 1958 paper. As always, their bogus 1937 paper was based on a bogus 1935 paper by J.R. Beach and O.W. Schaum called A Filterable Virus, Distinct from That of Laryngotracheitis, or what singers call losing your voice for several weeks from overexertion and muscular exhaustion, the cause of respiratory disease of baby chicks, who never stop chirping loudly, despite their untrained vocal cords, 24/7 for days on end. Apparently, in the case of IBV or infectious bronchitis virus, both strains of the disease could be regularly transmitted to healthy baby chicks by injecting into their noses or throat trachea exudate, meaning a substance that has oozed forth, or what we'd call the phlegm or boogers from the respiratory passages of diseased chickens. After being passed through a Birkefeld filter screen or strainer to get out larger particles like bacteria, but not the rest of the booger muck, and lo and behold, injecting sick chicken boogers directly up baby chicks' noses or in their throat trachea gave some, but not all of them, dyspnea, or shortness of breath, gasping and coughing, proving it was a virus and not just waterboarding newborn baby chicks with sick chicken boogers. Injecting mucus into a baby chick and making it react with shortness of breath, gasping, or coughing does not prove an infectious particle in that mucus made it react that way, and not all of the other toxic debris from our bodies that is found abundantly in and removed naturally via our mucus, or the unnatural introduction method of that mucus via injection for that matter. Ironically, the real danger to humanity is not the imaginary particles that virology claims to be protecting us from, but that the fictional coronavirus genomes that have been templated out of the virologist's false speculations are now used as templates to create and inject real products into hapless recipients who were conned and gulled into believing that virology's latest invention was real. That is, virology's fictional genomic inventions have been relied upon to create wholly unnecessary medical and political interventions. The dangerous and highly experimental mRNA and nanolipid biotechnology has killed more people the last 3 years than all other vaccines combined over the last 30 years, and we have only just begun counting. Sadly, the average virologist really believes they are helping humanity, clueless that there is nothing to protect us from except their own products. Agencies like the U.S. Center for Disease Control are also propagating this madness, and the CDC took 8 months to respond to a Freedom of Information request surrounding their claim of isolating SARS-CoV-2 in their June 2020 Emerging Infectious Diseases publication, Severe Acute Respiratory Syndrome Coronavirus 2 from Patient with Coronavirus Disease, United States, by Jennifer Harcourt et al. The questions that were put to the CDC were simple and included the following. Did the scientists for this paper use control groups? If so, did the control groups use the same formulations of cell culture mixtures as the experimental groups, sans the sample containing the alleged viruses? In summary, if control groups were used, please list the details of the control groups. The reason she's asking is that we know for a fact through virologist Stefan Lanka's control experiments that these toxic antibiotics they use in their cell cultures cause the same cytopathic effect breakdown as the alleged viruses they claim are doing it. And the cell culture itself has genetic material from the viro cells and fetal bovine calf serum that get used to assemble the in silico virus genome, even though they aren't alleged viruses. So you can easily cheat a control experiment by using 1 times or less of those toxic antibiotics in the control sample than the real one, or by leaving some or all of them out altogether and using a saline buffer solution instead, or by reducing or withholding the fetal bovine calf blood serum, which you can see keeps the cells alive and healthy longer, as opposed to reducing it to 1% and seeing cytopathic effects from just reducing that alone, or by not letting the control sample break down for the same amount of days as the original sample. Leave some meat out for 1 or 2 days compared to 5 days without any alleged virus or doing anything to it and see what happens. That's cytopathic effect breakdown. Strangely, instead of asking Jennifer Harcourt, a microbiologist who actually works for the CDC, or one of her team who actually wrote the paper, to answer this simple request, instead, on March 29, 2022, the CDC responded that they had located 37 pages of responsive records and 1 Excel spreadsheet, allegedly pursuant to the material requested. In summary, the CDC's responsive records included the following: internal CDC emails sharing images such as this, purporting to show scope pics of potential 2019 nCoV from the first U.S. case. Like this pic of a potential unicorn isolate in some guy's yard. CDC research microbiologist Azaibi Tamin hoped some of these 7 lysates, or cellular destruction, showed the cytopathic effects are caused by the 2019 nCoV. Sounds conclusive to me. Rashi Gautam had tested 4 extracts, but all 4 extracts had tested negative with all 8 master mixes. Virus confirmed. While Stephen Lindstrom commented they were very nice unhappy cells. The respiratory virus immunology team lead, Natalie Thornburg, then asked if they could send the original JPEG or TIFF picture files for their cytopathic effect images. I want to start working on a publication quality figure. In their entire 37-page FOIA response, the details of the mock experiments were not provided by the CDC, despite being specifically requested. All that was sent was more than half covered up pictures of the mock experiment slides behind the other slides that are supposedly evidence of the cytopathic effects and thus implied existence of SARS-CoV-2. A bit like saying the damage to the ground in this photo proves the existence of unicorns. They then claimed it was legit by including GenBank accession numbers MTO20880 and MTO20881, which were listed in the Harcourt et al. CDC publication and already publicly available without need of a FOIA request, even though you could use these same bogus techniques on a horse's bottom, submit it to GenBank as a novel unicorn genome, and future genealogists could claim Clydesdale horses are unicorns because they can make an 81% genetic match out of their ground-up bottoms to your GenBank unicorn genome. Then, strangely, rather than answering details on what Jennifer Harcourt's team did in their study as requested, the CDC instead attached a summary copy of the NAZU New England Journal of Medicine paper, a novel coronavirus from patients with pneumonia in China 2019, which according to CDC electron microscopist Cynthia Goldsmith has two very nice electron microscopy images in Figure 3, one from human airway epithelial, referring to this. The usual black arrows pointing to particles of unknown provenance or genetic makeup and claiming they are 2019 nCoV virus particles without giving any proof. In fact, when Virus Mania co-author Torsten Engelbrecht asked multiple study authors, "Do your electron micrographs show the purified virus?" Dr. Wenji Tan from the NAZU team responded, "We show an image of sedimented virus particles, not purified ones." Well, how do you know that's a restaurant and not one of the thousands of other kinds of businesses in the shop fronts in that area? Is that a picture of sedimented restaurants because there must be some restaurants in that area somewhere? Had the CDC bothered to read the methods section of NAZU's paper, they would have seen it is full of the same bogus, nonsensical procedures this film has covered ad nauseum up to this point, as there is no part of the paper that demonstrated the composition or biological function of these imaged vesicles. If you want to prove or claim that's a restaurant and not a dry cleaner's, you have to go in there and show there's a commercial kitchen inside, and not just steam presses and hangers. They also included this completely blank worksheet. I mean, what more proof do you need? They did at least supply a page starting with, "For administrative convenience and to fully respond to your request, program staff have provided the following information below with corresponding web links," which unfortunately provided absolutely no information related to how the CDC's viral isolation experiments were suitably controlled. On December 23, 2021, Christine Massey also submitted a request to the CDC seeking full details of the Harcourt et al. Mock infected experiment, including the quantity of material from uninfected nasopharyngeal and oropharyngeal swab specimens that was added to the cell culture control group. The CDC eventually responded to Massey's request 4.5 months later, on May 10, 2022, with 36 pages of similarly unhelpful information, and the excuse that, in regards to certain portions of your request, a search of our records failed to reveal any documents pertaining to your request. These portions relate to your request for specific cell culture experimental group details and cell culture mock infected control group details and whole genome sequencing purity and control details. Your request was sent to the National Center for Immunization and Respiratory Diseases, NCIRD, for search. They responded that certain details in your request were not available as records controlled or maintained by the CDC. In other words, the CDC appear completely ignorant to the fact that they are not following the scientific method, or they have realized that the game is up and are engaging in disingenuous responses. like purposely withholding revealing information under Exemption 6 because it might also contain personal information such as a cell phone number. Well, that didn't seem so hard. Either way, the CDC cannot be taken seriously as a source of reliable scientific information if they are also promoting uncontrolled experiments as proof of viruses. Now that you understand more about virology than the $116,000 a year public tax-paid microbiologists like Jennifer Harcourt at the U.S. Center for Disease Control, let's circle back to the February 2020 Peng Zhou paper in Nature, a pneumonia outbreak associated with a new coronavirus of probable bat origin, claiming the identification and characterization of a new coronavirus 2019 nCoV. In their alleged isolation experiment, the authors produced images showing apparent cytopathic effects after 24 hours in the alleged 2019 nCoV infected Viro E6 monkey kidney tissue cells, but no cytopathic effects in the mock infected cells, the latter purported to be a control. There you see it. The mock-infected cells look normal. The alleged 2019 nCoV infected cells are clearly showing cytopathic effects. Case closed, right? Wrong. Because aside from at least admitting the experiments were not randomized and the investigators were not blinded to allocation during experiments, nowhere in the methodology section did they provide details on how they did these alleged mock infection control experiments. Until when questioned later in August 2021, when co-author Xing Lao Yang revealed some startling admissions. Firstly, aside from the fact that there were no positive control experiments, meaning with comparable human samples from healthy people minus the alleged virus, treated the exact same way as samples from sick people assumed to have it, Yang stated they doubled the dose of penicillin and streptomycin in the experimental group, giving the control groups only 1% of both antibiotics. And then giving the alleged viral comparison groups 2% of both antibiotics, and then an additional 1% again on top. But we know for a fact that these two antibiotics cause the exact same cytopathic effect breakdowns themselves with no alleged virus needed. So is it really any wonder these are the visual results they got from not actually controlling their own control experiments? You may as well hit your control group's finger once with a hammer and then hit your other group's finger twice or even a third time for good measure and claim that's proof smashing your finger in a door is guaranteed to break it. Yet, when asked why this variable was altered, the response was the intention of anti-anti, the two biotics, is to prevent contamination from bacteria or fungi during virus isolation. So 1 or 2% concentration did not affect the cell growth. 2% in the first generation was just to prevent contamination from the samples. So you lost your businesses, your personal liberties, and your beloved grandparents because trusted experts like Yang do not understand the meaning of a control experiment where all variable factors must be kept constant due to his purposely taught indoctrination.
But he can't be blamed. He's only a pawn in their game.
It was then suggested to Yang that they should run the control experiment again with the same higher 2% dose antibiotics exactly as they did for the other wells to scientifically ensure that this was not in fact one of the factors inducing cytopathic effects in the kidney cell line. Yang subsequently provided the evasive response: if you could make sure that you could prevent contamination from bacteria or fungi, you do not need to use the anti-anti, seemingly ignoring the crucial point that it could be the additional antibiotics themselves that were toxic to the cells. Particularly as streptomycin is known to be nephrotoxic, causing formidable nephrotoxicity according to the Mayo Clinic, and called a potentially nephrotoxic agent by the chemists and biologists on the NIH database, causing excess cellular excretion, or what Yang calls viral cytopathic effect, producing cellular debris particles virologists call viruses. Yang says it doesn't matter. Anti-anti has no effect for cell growth, while ignoring the fact that it causes the cell destruction they are looking for. Either way, altering these second antibiotics variables on top of the varied source materials makes these control experiments unscientific, meaningless, and thus invalid. Another staggering, or rather shocking, revelation from Yang was that in their experimental groups, each group had 24 different tissue culture wells, yet only one out of 24 wells containing Vero E6 green monkey kidney cell cultures showed any evidence of cytopathic effects, and only one out of 24 wells containing HUH7 cells taken from a human liver tumor in 1982 showed any evidence of cytopathic effects. To put this absurdity visually, 48 out of 48 uncontrolled control experiments on 2 different cell culture tissue types with only 1% antibiotics and no alleged viral material added looked like this. And 46 out of 48 of the cultures with 2% antibiotics that allegedly had viruses added to them that break down tissue with cytopathic effect also looked exactly like the ones that didn't. Sorry to be your yin, Dr. Yang, but you don't need a biology background to understand blatant charlatanism when you see it. Again, from part 1. If you only had a 2 in 48 chance of your parachute opening, would you trust the science enough to jump? Clearly virologists do, because what should be considered an experimental margin of error is the basis of one of the declarations of a claimed deadly new pathogen described in an article that, as of November 2023, has been accessed 1.45 million times and cited in other papers and publications nearly 13,000 times. Do the other authors who are citing this paper realize the flimsy gossamer of evidence that this house of cards called COVID-19 is built upon? Perhaps they would not even be bothered by such a revelation, as actual biological experiments are being increasingly abandoned, while man and his computer-created in silico genomes are absurdly claimed to provide adequate evidence for the existence of viruses. In the case of Peng Zhao et al., their computer simulation was proudly proclaimed to be 96% identical at the whole genome level to a bat coronavirus. They decided to template their new viral invention against this bat sequence based on the nonsense that previous studies have shown that some bat SARS-CoV-2s have the potential to infect humans. Their computer software assembled what became GenBank accession numbers MN996527 and MN996532 from this form of faux evidence, which also lacks valid controls. Yet, every virologist in the world can now computer assemble your boogers into either of these fictional genomes and claim, oh my God, you must have the Batman virus. In early 2022, a mathematician working with virologist Dr. Stefan Lanka released an analysis of the associated sequence data produced by Fan Wu et al. Startlingly, it was concluded that a repeat of the de novo assembly, or putting together the exact published short genetic contig sequences by Fan Wu's team, taken from lung sputum, and trying to get the computer to assemble them into an already existing virus genome from the database using megahit version 1.2.9, showed that the published results could not be reproduced. We may have detected ribosomal ribonucleic acids of human origin out of human lung sputum? You don't say, contrary to what was reported by Fan Wu et al. Evidence is lacking that only viral nucleic acids were used to construct the claimed viral genome for SARS-CoV-2, and not actually the total combined multi-source genetic abundance soup you find in a lung sputum sample. Further, with respect to the construction of the claimed viral genome strand, no result of possible control experiments have been published by Fan Wu. Surprise, surprise. This is equally true for all other reference sequences considered in the present work. In the case of SARS-CoV-2, an obvious control would be that the claimed viral genome cannot be assembled from unsuspected RNA sources of human or even other origin. Meaning at least try the exact same procedures on samples from a healthy human, or even a banana for that matter, and make sure you can't assemble the exact same alleged viral genome sequence out of those two. It doesn't matter how many thousands of monkey see, monkey do's can cite a bogus experiment like Fan Wu's in their own reference sections without even bothering to double-check its methods, or how many scientific advisors cite those thousands of monkey see, monkey do's citing that bogus experiment as proof, if the actual scientists trying to repeat the experiment aren't able to get the same results and confirm it as valid as the scientific method demands. Indeed, this independent analysis only obtained 28,459 contigs, significantly less than the 384,096 described by Fan Wu et al., meaning there were 356,309 missing assembled genetic sequences, or nearly 93% short of what was claimed to be there, if you want to get nitpicky about it. So where did you get those, Fan Wu? Additionally, the longest computer-constructed contig independently obtained was only 29,802 nucleotides long, which was 672 nucleotides shorter than Fan Wu's longest of 30,474 nucleotides. Like Subway, claiming this is a foot-long sandwich, meaning the published sequence data from Fan Wu et al. Cannot be the original reads used for assembly. Which means Fan Wu's team is either grossly incompetent at their jobs or purposely publishing lies. The independent analysis also concluded that alignment with the nucleotide database on 5/12/2021 showed a high match, 98.85%, with human Homo sapien RNA.
Have you ever seen a Homo sapien?
You don't want to know. Yeah, I saw one once at the zoo.
This 98.5% Homo sapien RNA match was to pre-ribosomal N4 RNA 45SN4 ribosomal RNA found in GenBank on 4/7/2020, which also just happens to be found on human chromosomes 13, 14, 15, 21, and 22. But if genomic nucleotide sequences allegedly determine the unique makeup of different organisms, like a human from a bacteriophage, and this alleged virus allegedly came from a bat first and later jumped to humans, then how is its genetic sequence made out of common human sequences if a virus isn't human and didn't even come from humans? Obviously, because Fan Wu's team assembled the fake virus genome out of chopped up bits, including the human lung sputum sample. It's a smoking gun. This observation contradicts Fan Wu's claim that ribosomal RNA depletion was performed and the human sequence reads were filtered out using the human reference genome, human release 32 GRCH38.P13. If you filtered out the human reads, then why are they still in your virus genome? Of particular note here is the fact that the human ribosomal RNA sequence NR146117.1, dated April 7th, 2020, was not even published until 2 months after Fan Wu's publication of the SRR10971381 sequence on February 3rd, 2020. So how did they filter out a human reference genome read that didn't even exist in the database yet? And again, given the high error rate, it raised the question of whether some of the sequences were simply those generated by the PCR amplification conditions themselves. So, in their independent study, virologist Dr. Stefan Lanka and his mathematician took the original published genomic sequence data for the original work on coronavirus SARS-CoV-2 from Fan Wu's team, remember, based on the lung sputum fluid from a single patient in Wuhan, said to have only an 89.1% match to a group of SARS-like beta coronaviruses alleged to have been previously found in bats in China, and started running this one patient sequences in GenBank against other available reference sequences for not only things like bat SARS-CoV, but also other alleged virus genomes that have nothing to do with SARS, like HIV, hepatitis delta virus, measles virus, Zika virus, Ebola virus, and Marburg virus. And lo and behold, they were able to get anywhere from 82% to predominantly 96 to 100% matches to all of these other viruses from this same one patient sample sequences, much more than Fan Wu's 89% match to an alleged bat coronavirus. This must be one unlucky patient to have caught SARS-CoV-2, bat SARS-CoV, HIV, hepatitis delta virus, measles, Zika, Ebola, and Marburg all at the exact same time. Of course, the truth is, if the virologists want to find a virus, it all just depends on how they design their protocols and what they ask the computer to look for, and it will find it. And how would these fortune tellers know what to even look for? To sustain the illusion of the COVID-19 pandemic, cases were required. Look at that scary number on the Johns Hopkins dashboard. Over 676.5 million cases of COVID-19 in just under 3.5 years. That's just over 0.8% of the population, or just less than 1 out of 100 people tested positive or were diagnosed with COVID-19. And look, just under 1 out of every 10,000 people allegedly died from it. But did they really? These alleged cases were provided by the world's largest ever human testing program, involving billions of PCR kits from dozens of manufacturers distributed around the world. It remains unclear as to why the doctor in molecular genetics, Professor Stephen Buston, who is a world-renowned expert on quantitative PCR, whose research focuses on translating molecular techniques into practical, robust, and reliable tools for clinical and diagnostic use, failed to decisively point out the inappropriate use of the PCR process. And he should know better, as Stephen Buston was the lead author for the 2009 publication, the MIQE Guidelines, Minimum Information for Publication of Quantitative Real-Time or RT-PCR Experiments, in which the key conceptual considerations for RT-PCR experiments were outlined by him and his team as follows. Analytical specificity refers to the qPCR assay or test detecting the appropriate target sequence, rather than all of the other non-specific targets also present in the sample. Diagnostic specificity is the percentage of individuals without a given condition, meaning not sick with any symptoms, whom the assay also identifies as negative for that condition. A PCR molecular detection test, only designed to detect normal peanut fragments in a mixed snack bag if you dig deep enough to find them, can't tell you if you're going to have a deadly allergic reaction to those peanuts or not. It just tells you if there are peanut traces in that bag or not. If Buston had remained true to the science, then he should have called a halt to the PCR pandemic that turned into the COVID-19 fiasco before it even got started, back in January 2020, when the Corman-Drosten PCR protocols were published that enabled the whole nightmare. The word specificity appears only once in the Corman-Drosten paper, and it had nothing to do with diagnosing a clinical condition, let alone a viral infection. There was no detection of 2019 nCoV as the paper claimed. All that was established was the analytical specificity of their assay to detect selected target sequences of the published 2019 nCoV in silico genome. It was an in vitro molecular reaction experiment that they generated themselves with synthetic nucleic acid technology that does not require the existence of a virus. In fact, they admit there was no virus. In the present case of 2019 nCoV, virus isolates or samples from infected patients have so far not become available to the international health community. So their test was designed in absence of available virus isolates or original patient specimens by creating synthetic sequences that looked close to published sequences from the 2003 SARS-CoV sequences. You can't design a valid PCR test to detect something you don't even have possession of or know what to really look for in the first place. And unfortunately, all of these people bought this con job hook, line, and sinker. Furthermore, there was no establishment of how the PCR positive result related to a clinical condition or symptoms, i.e., the COVID-19 PCR kits were never shown to diagnose anything in a human subject. This PCR test created an invented new disease based on a fictional virus. What was not well publicized was that the world expert on PCR, Stephen Buston, admitted to David Crow already back in April 2020 that...
So what I've seen with the coronavirus testing is that they choose a cycle number. I've seen 36 and 37. I haven't seen it published very much. Yeah. And if you obtain sufficient DNA by that cycle, it's considered positive, and if you don't, it's considered negative. Yeah. Is there any? I mean, that seems kind of arbitrary.
It's absolute nonsense. Yes, it's absolute nonsense. Well, it makes no sense. It makes no sense whatsoever.
However, the PCR fraud was even more apparent when journalist Eric Coppolino interviewed Stephen Buston on Planet Waves FM in February 2021. Coppolino's intention at the time was to find out more details about the problematic reverse transcription RT step of the RT-PCR process, of which there are many problems, but he was stunned after the interview to realize that what he thought was a sometimes inaccurate test was completely fraudulent.
But that's not being done. Every time someone gets a PCR positive, the New York Times and everybody else is calling that a confirmed case of SARS-CoV-2, of COVID.
Yeah.
And then they're saying there's like 250,000 of these a day. I don't know a single person who's sick.
You don't?
I don't.
Well, we have our ICUs are overrun at the moment.
So Buston nervously evaded the PCR issue by claiming ICUs are overrun at the moment, rather than admitting the diagnostic specificity of the PCR kits had never been established.
It was the last straw for his frustrated surgeon, Dr. Craig Urisevich, who last night lashed out on social media, sharing photos he says paint a damning picture. A 12-bed ICU ward sitting idle, 6 operating theatres empty, some used as storage rooms. One's even become the backdrop for a film crew instead of firing up for surgery.
Buston then further defended the PCR protocols in use with the assertion that
I'm not quite sure what the problem is, because what was obvious was that the disease, this pneumonia, was being caused by this virus, and this virus started cropping up in all the places where more and more people were coming down with the same symptoms, and these primers were detecting that virus.
Well, the problem is, Stephen, these people were getting the exact same symptoms everyone has always gotten during flu detox season since time immemorial, and nothing on this symptom list is new or special. And these primers can't detect a virus because the sequences they are looking for weren't taken from a virus. They were computer assembled from normal genetic material into a virus.
The idea of, true purification, you separate it into centrifuge and you've got of only that. Mm-hmm. And then you can, then that is the thing that is sequenced and then used to prime the PCR. It does not appear that's what's happening.
Well, the way the sequence was established was by taking the sample from the original patients, growing up something, and then sequencing it, and then just assembling the sequence. And what came out of that was a SARS virus.
And here we have the crux of the problem. As David Crow said following his own interview with Stephen Buston, scientists like Buston have a tendency to assume that everything outside of their domain of interest is true, and they can rely on it. Had Buston read Fan Wu's full paper and examined the methodology, he would have known his own PCR test may as well be looking for dog turd sequences rather than Fan Wu's published sequences, because neither of the primer sequences provided proved the existence of the alleged SARS-CoV-2 virus, no matter how well your PCR test can detect small sequences from either accused target. The crucial issue is that it doesn't matter how well designed any primers are if the provenance or origin or the significance of the genetic sequences being amplified through the PCR are unknown, then nothing more can be concluded by their mere presence. Buston can reassure the world all he wants about the potentially very high analytical performance of a PCR protocol, but the establishment of its diagnostic performance is where the rubber meets the road. Even if SARS-CoV-2 had been shown to physically exist and the PCR was accepted as a valid diagnostic tool, Buston would have to admit that none of the PCR assays have been developed as his MIQE guidelines specify, and none qualify as being clinically validated. Your PCR test could be 70 to 100% accurate at finding partial chocolate cake fragments in my mouth, but it can't tell me eating that cake made me sick. It was a surprise that earlier during the same interview, Buston denied any prior knowledge of the false pertussis outbreak in Dartmouth Hitchcock, New Hampshire in 2006, when the PCR kit that was rolled out resulted in a 100% false positive rate. Buston claimed to have learned about it for the first time just days before that interview, some 15 years after the incident, when he read about it on Coppolino's website from an article provided for the purpose of the interview. Yet the incident was very well known and received coverage in the New York Times, with comments from many public health and diagnostic test professionals. By 2006, Buston was a professor of molecular biology at 2 different universities, so it's a small wonder that the PCR specialist had not had any inquiries from medical colleagues back in 2006 when the incident happened. Buston would have been one of the very few PCR experts even in existence at the time to contact, and it was an early indication of how the PCR could be catastrophically misused as a clinical diagnostic tool, where a Dr. Kretzinger admitted, "We have had a number of outbreaks where we believe that despite the presence of PCR positive results, the disease was not pertussis." If that wasn't bad enough, it related to an incident where the purportedly causal microbe, the bacterium Bordetella pertussis, is something real that can be physically isolated, genetically and chemically characterized, and sequenced, and its genetic sequences confirmed for the PCR to be calibrated against, and it still failed miserably. Whereas in contrast, the SARS-CoV-2 PCR protocols are simply calibrated to genetic fragments of unknown origin from human lung sputum. By the time Buston was interviewed by Coppolino, he had already co-authored and submitted a paper titled COVID-19 and Diagnostic Testing for SARS-CoV-2 by RT-qPCR, Facts and Fallacies, that was published later in February 2021. In this paper, Buston and co. stated that the Corman-Drosten assay worked and was specific and demonstrated astounding sagacity, meaning sound and reason, and selflessness by the scientists involved, as well as the remarkable speed with which PCR-based tests can be developed and put into practice. Specific for what? And sound and reason how? We already saw the Corman-Drosten paper made fake synthetic sequences based on a blind guess from SARS-1 16 years earlier because they had no virus isolates or patient samples to even design a test for SARS-CoV-2. The obvious question remains, specific for what? Were Buston and co. implying that the PCR tests are specific for short-targeted RNA sequences, a coronavirus known as SARS-CoV-2, or the WHO-invented disease known as COVID-19? The Corman-Drosten paper only established the analytical specificity for amplifying some selected RNA sequences. It had nothing to do with the establishment of a virus or diagnosing a disease because it admitted it had no virus isolates of SARS-CoV-2 nor samples from a patient specimen to determine any clinical COVID-19 disease. So Buston, the developer of the MIQE guidelines, surely knows that of the 3, only the first was scientifically established and nothing was or has been validated for clinical application. And yet, his paper goes on to make the ridiculous, illogical claim that PCR testing is highly suitable for large-scale testing, as demonstrated daily by the millions of tests carried out to date. And Monsanto's Roundup is safe to consume, demonstrated daily by the millions of crops it is sprayed on. Has world-renowned PCR expert Buston Forgotten that the tests are simply a molecular amplification tool and not for diagnostic use?
I don't think you can misuse PCR. No, the results, the interpretation of it, it doesn't tell you that you're sick, and it doesn't tell you that the thing you ended up with really was going to hurt you or anything like that.
That's because the PCR simply amplifies invisible genetic sequences through molecular reactions until there are millions and millions of copies to be able to detect that small sequence. Even if the PCR protocol is performed correctly and has a known 100% analytical sensitivity and specificity, not too many cycles to create false sequences that aren't in the sample, etc., the molecular reaction itself has no capacity to determine the origin of that genetic sequence or the relevance of it being there. A positive result does nothing more than confirm the presence of a molecule sequence it was looking for, not what that sequence is or what it does. However, if claims are being made that the PCR is a diagnostic tool, like the Mayo Clinic website, it should be obvious that clinical validation studies would need to be performed before the test was introduced into clinical practice, meaning clinical evidence there is some new, unique disease this test claims to be detecting. The Corman-Drosten paper skipped this clinical validation step, and the World Health Organization accepted the fraud by placing versions of the PCR protocol on their website on the 13th and then 17th of January 2020, before the paper had even been published. After that, the PCR was simply used via circular reasoning to make claims about diagnosing infections in hundreds of millions of people, even if they had zero clinical symptoms. The next phase in the early stages of the alleged pandemic involved experts, such as Australian infectious disease specialist, Associate Professor Sanjaya Senanayake, promulgating unfounded claims about the accuracy of the test to the public.
That's a hard one to know, Jeremy, because there's no gold standard to really compare this to. So if we had a new test for picking up golden staph in blood, we've already got blood cultures. That's our gold standard we've been using for decades, and we could match this new test against that. But for COVID-19, we don't have a gold standard test. So the current test we are using, the PCR test, where we put a swab in the throat and in the back of the nose, they're our gold standard. But trying to work around that, we think that it's probably picking up about 70% of cases.
A gold standard means an already known and reliable test to check a new test against to make sure it's also just as reliable, like an ultrasound with a visible fetus in it, or a giant kicking lump in a woman's belly, would both be good gold standards to check the accuracy and validity of a new pregnancy test against. If this new test came out negative for these two mothers, you'd know it's a useless pregnancy test. Senanayake implied that if you don't have a gold standard to check a PCR test validity against, you can just assume that a new PCR test can validate itself. If a PCR test told 30 out of every 100 women like this that they probably weren't pregnant, would you trust the science enough to make that a gold standard?
We think that it's probably picking up about 70% of cases.
At least he was telling the truth when he said,
there's no gold standard to really compare this to.
Because the real gold standard is something that doesn't exist, that being the physical isolation and proof of a viral particle. The World Health Organization were clearly not concerned by the lack of a gold standard or any evidence of a virus, and cemented the PCR fraud by stating that a COVID-19 case, not a suspected case, not a probable case, but a confirmed case was, conveniently hidden and mentioned last at the bottom, a person with laboratory confirmation of COVID-19 infection, which in 2020 was typically PCR, irrespective of clinical signs and symptoms. In this one sentence, they proclaim that the clinically unvalidated PCR tests have 100% diagnostic specificity and nonsensically twist the meaning of the word infection to include individuals who have no signs or symptoms. The etymology of the word infection provides a derivation from the Latin inficere, meaning to spoil or to stain. Mosby's Medical Dictionary, 2009, states the definition of infection to be 1. The invasion of the body by pathogenic microorganisms that reproduce and multiply, causing disease by local cellular injury, secretion of a toxin, or antigen-antibody reaction in the host, and 2. A disease caused by the invasion of the body by pathogenic microorganisms. The established meaning of infection relates to a disease state of local cellular injury caused by the alleged virus or microorganism. A diseaseless state of disease is an oxymoron. Otherwise, a term such as commensalism should be used instead, meaning a relationship between two kinds of organisms in which one, like microorganisms, obtains food or other benefits from the other human or animal organism without damaging or benefiting that human or animal. The WHO invented an absurd new definition of pandemic, excluding references to the words with enormous numbers of deaths and illnesses. Apparently, you don't need to be sick or die anymore to be in a dangerous pandemic. So now even the seasonal flu could be classified as a pandemic, and they are now subverting the definition of infection, one that disconnects it from the concept of disease through the sole use of PCR results.
PCR is separate from that. It's just a process that's used to make a whole lot of something out of something.
Unfortunately, on more than one occasion in the COVID-19 era, influential figures such as Buston and Senenanyake have supported the virologist's use of a molecular manufacturing tool to make all sorts of unfounded claims, including both the unratified ability to diagnose a novel infection and the detection of an alleged virus. Of note, a biased misinterpretation of the PCR appears to begin before the amplification process has even started. For example, the Roche high-pure viral RNA kit used to prepare samples for the PCR states that it rapidly isolates viral RNA from mammalian plasma, serum, body fluids, and cell culture supernatants. Yet it's unclear from the supplied product information how the kit would possibly separate alleged viral RNA from all of the other RNA present in the sample. The process itself includes a poly-A carrier RNA binding additive step. The problem is, polyadenylated sequences are non-specific and interact with cleavage factors from most tissues. Despite all of the binding, buffering, and centrifugation steps they describe to isolate the alleged viral RNA, this would still not be able to differentiate the origin or source of the RNA they isolated. Despite this, the protocols section proclaims that the end product is purified viral RNA. Anyone believing this unfounded claim then thinks their subsequent positive RT-PCR result is evidence of a virus, and not just regular RNA from God knows what found in many people's lungs, throats, and noses. The same can be said for Roche's high-pure viral nucleic acid kit, used by teams such as Nazhu and Pengzhou in their claims to have discovered SARS-CoV-2 in patient specimens and cell culture experiments. Once again, Roche makes the dubious statement that the steps outlined in the protocols section would result in purified viral nucleic acids. Incidentally, Buston was asked via email specifically about Roche's claims when the following was put to him. I assume the kit must be able to distinguish viral nucleic acids from all the other nucleic acids in the sample. Do viral nucleic acids have a chemically unique property from the others? Buston responded, the extraction process is not specific for any particular nucleic acid, but it can be specific for types of nucleic acid. Some kits can differentials extract DNA or RNA, but this means any DNA and RNA will be present in the extracted sample. A small amount of the extracted material is then subjected to the PCR reaction. This is what provides the specificity. In plain English, Buston did not attempt to provide an explanation for Roche's fraudulent claims that their kits can isolate viral nucleic acids for PCR or RT-PCR, but purposely tried to confuse or obfuscate the issue by substituting the specificity of provenance or the origins of the nucleic acids with the specificity of the sequences being selected for the PCR, no matter what source they came from. Again, not to beat a dead pseudoscience to death, but while PCR can look for any specific sequences you tell it to, PCR cannot tell you those sequences came from any such thing as a virus in the sample. This amounts to a linguistic sleight of hand that helped allow a virus to appear out of thin air. In Part 3, Gaslighting, the lab leak distraction, virology and the closed society, and metagenomic sequencing, virology's final gasp.